I am assistant professor of entomology at the University of Florida, entomology and nematology department. And I am located at the Southwest Florida Research and Education Center here in Immokalee, Florida. I will be presenting the work that we have been doing on citrus pest management and our efforts toward developing integrated and more sustainable pest management programs. Citrus in Florida and several parts of the world is colonized by a wide range of pest groups. I have placed some of them here, which are more important, and particularly from our Florida perspective. So we have Asian citrus hillet, several species of scales, aphids, mealybugs, whiteflies, and stink bugs and leaf-footed bugs among the second groups. Then we have also rust mites and spider mites, and then also others like citrus leaf miner, thrips, fruit flies, and weevils. Since its introduction in the United States, which was here in Florida in 1998, the Asian citrus hillet basically dominates the whole pest complex because of its association with the devastating Huanglong Bing or citrus greening disease, which is highly significant for Florida citrus industries and reducing productions significantly over a decade now. So basically, most of the efforts are focused toward this pest. So most of my talk will also cover our efforts with this pest. So I will give a brief introduction to the pest, its association with the disease, and then I will talk about biological, chemical, and cultural methods of its control and the different directions my lab is working in. This is the adult of the Asian citrus hillet. It's a small insect, about 2 millimeter. You will see the adult sitting at this 45 angle. They are capable of laying several hundred eggs during their life. Studies have shown up to 700 and 800 eggs potential. They go through 5 nymphal instars, and then they become adult. So the whole life cycle is completed in 3 to 4 weeks, depending upon the temperature. So basically, it's the adult that is responsible for carrying the pathogens and spreading from the tree to tree and grove to grove. And that's one of the primary source for the spread of the disease. Obviously, the nymphs, they are capable of acquiring the pathogen and contribute to its multiplication, but eventually it's the adult that takes it from tree to tree and grove to grove. Here are some symptoms of the disease, the blotchy model on the right side on your screen. And then it's also sometimes confusing with some of the micronutrient deficiencies. But as you see here, this blotchy mottling across the veins is very peculiar, and you can easily identify it from other nutrient deficiencies. It was identified from Florida in 2005, where the psyllid, I had mentioned on the previous screen that it was identified in 1998 from Florida. Once the disease starts to develop, that's the typical symptom. You will see a yellow shoot developing someplace in the canopy. And that's where the name Huanglong Bing in Chinese come as it relates to the yellow shoot. So that's how the vector or the Asian citrusillid interacts with the pathogen. Just a brief summary of the events. First of all, the transmission, it could take place within 1 day to the shoot by the adult ACP, which you see here. And then it takes about 15 days for acquisition and amplification of the candidatus Liparibacter asiaticus by the nymphs. And the emergence of infected adults, as I said, the life cycle is about 3 to 4 weeks. Within 30 days, the infected adults will be emerging and spreading the disease. So incapacitation of phloem within about 3 months, and then it starts to infect the root system within 5 months, and you also start to see the foliar symptoms showing up as well. And generally speaking, there's a time lag, or depending upon what level of pest infestations you have on your trees and in your groves, the symptom appearance could take from 6 months to a year before you start seeing them. So as I said, the Asian citrusillid, it's a primary vector. It's spreading between trees, the pathogen. And you have 2 adjoining trees here. You see a lot of fruit drop, which is one of the symptoms of the Huanglong-Ping disease. And the neighboring trees, you don't see that much fruit drop, but it's only a matter of time because disease is already spread in these neighboring trees as the vector was moving back and forth between these trees. So it starts to happen soon. And finally, it's very devastating. And then the fruit size is usually small when you have the infection, and the fruit quality, the fruits are lopsided, and you see the aborted seeds, and these are, and even the taste will be bitter once you taste the fruit. So these are some of the symptoms of the HLB or Huanglong-Ping disease. It's also called citrus greening disease. This slide just shows you how it's infecting and impacting the Florida citrus industry. As I mentioned earlier, that the vector psyllid was identified in 1998, and the disease was identified in 2005. And there were other events in the previous years which impacted the citrus production to some levels. But here, once the Huanglong-Ping disease was identified, then you see a consistent decline in the production. And we have lost more than 50% production over the past 13 to 14 years. So from that perspective, the management of the vector psyllid is critical because of its association with the pathogens of Huanglong-Ping or citrus greening disease in order to reduce the incidence and the severity of HLB, to sustain the mature trees, bearing trees which are already there, and to protect the young trees. If the vector is not managed carefully, you eventually end up getting to the point where you need to remove those trees, blocks and blocks of citrus. This is some of the history or the different things that were recommended in relation to managing this vector pathogen complex. Initially, it was started that we should be greenhouse. We should have greenhouse covered with psyllid-proof screens for plant production, mainly for the budwood production. Now we also have projects where we are taking this tactic and trying to produce citrus, especially the fresh fruits, in these protective houses with ventilated screens so that we can isolate them from the vector as well as for the pathogen. But the vector control in the open production systems is critical and highly emphasized because you cannot get successful citrus production without vector control. Initially, it was economical to take out the symptomatic trees, but then the disease spread through the state quickly, and now it's not viable because most of the trees are infected. So we are obliged to learn to live with the greening and use as many tools as we can to reduce the spread of this vector pathogen complex. Some of the concerns, obviously, as I said earlier, that most efforts are focused on the Asian citrus psyllid and HLB for their management, but still the incidents keep growing. And the other concerns that we have for the pests and diseases are citrus leaf miner, which is associated with the citrus canker disease, different species of mites, rust mites, spider mites, broad mites, false spider mites associated with leprosis, aphids, different species, brown citrus aphid, green aphid, or spirea aphid, brown citrus aphid associated with citrus tristeza virus, then different species of scales and weevils. Pest resistance is another concern which we have seen, and it's happening due to the extensive use of insecticides, particularly neonicotinoids, and the cost of pest protection and production is going up. While we go with aggressive programs, high use of insecticides, we see the concerns of diminishing biological control, and then environment and health are also negatively impacted. So as we manage this vector pathogen complex, there are some short-term objectives and the long-term objectives that we need to keep in mind. For the short-term objectives, we have ACP control in mature and young trees. Then we have multiple habitats. We have conventional production systems relying heavily on the synthetic insecticides. And then we have organic production systems. And then we need to reduce the disease incidence in all these production systems. And even in the urban environments, and then the backyards, where people have a good amount of citrus trees as well. When we look into the long-term objectives, obviously we want sustainable agriculture and horticultural enterprises. We want to conserve the resources, both in terms of the effective mode of action insecticides. We don't want to lose them to resistance, and also biological control, which used to be and still is a very significant component of citrus pest management in Florida and even in California and other places. So we want to have economical and environment-friendly approaches to the pest management. So these are some of the things that help us with some of the basics as we develop the management programs. We know that the Asian citrus psyllid utilizes the newly developing buds and shoots to develop and reproduce. They need those shoots to lay eggs, and then the nymphs and adults need to develop on those soft tissues as well. This new growth also attracts beneficial organisms and other pests as well. One of the weakest links that we know is the overwintering of the Asian citrus psyllid during the time between November into February. That most of the mature trees in Florida are not producing the new growth which the psyllid requires to reproduce and develop. So that is the time when mainly the adult populations in low numbers exist. So that is the time if we can target them with insecticidal sprays, we can reduce population significantly before the season starts. And at that time, most of the beneficial insects and most other pests are not common. So you are not really doing any collateral damage to the beneficial fauna. So the different tools that we have, and some of which I will talk about today for vector management, for developing integrated pest management. We have biological control, chemical control, in which we have foliar sprays, as well as soil application of systemic insecticides to young trees, mainly neonicotinoids. And then we have culture controls, such as metallized plastic mulch. And I think we can also include those citrus under protective screens, which I showed you that we are doing some work on producing citrus in those enclosed structures, is also one of the cultural methods of pest control. And obviously, horticulture practices to mitigate the impact, which include nutrition, irrigation, soil management, are required and happening as well. And also research going in those, improving those practices as well. In terms of biological control, the lady beetles is one group, which is shown as highly significant in Florida system in several studies, including several of the previous people who worked on it and myself. And they provide significant reduction in the Asian citrus populations, as well as they also target other pests like citrus leaf miner, aphids, mites, scales, mealybugs, and several others. And you see here these different species on psyllid colonies feeding on the psyllid nymphs. This picture shows you the larva of one of the species called Olavi nigrum. And you see here in the picture on the left, it is feeding on a mature nymph of the Asian citrus psyllid, whereas picture here on the right, you see it feeding on the larva of Asian citrus psyllid. These beneficials, like I said, they are also useful for several species of pests, and therefore we need to conserve them and augment their population. And this slide, just to show you that how it is effective even against Florida red scale, which is covered by this hard armor covering on the body, which even sometimes is good enough to prevent the contact of insecticides with the mature scales inside or the newly developing crawlers. So this predator, we did some studies, and you will see that how it's able to go and remove the armor and feed on the females as well as on the crawlers inside those bodies. So this was very interesting behavior that we observed for this species, and it was never studied before. So we did several experiments of choice and no choice, and it was very obvious that they were very effective on the scale populations that were covered with the armor, suggesting that it could be a good predator for these scale insects. And then there are different species of Chrysoperla and Serocrysa that we have, and those are also very effective predators of psyllids as well as other species. So we did some studies a couple of years ago just to measure the impact of these beneficial insects on the populations of Asian citrusillid. And this graph basically shows you how the psyllid populations will increase if these beneficial insects were not there. And for that situation, we covered those colonies with these sleeve cages. So those were protected from the attack by the predators. And you see these light colored bars, the population of Asian citrusillid increasing in those colonies and in those trees. And the dark colored bars here, you see that most of those populations were taken out by the predators, showing a high impact of biological control. In some cases, 100% of those colonies were wiped out by these predators. So it definitely shows that if provided a chance, biological control and these predators are very effective against the psyllid populations. Obviously, when you have a vector that is responsible for a devastating disease, so you cannot rely just on one control tactic, and you definitely have to take other methods of pest control, particularly chemical control, into consideration. We do have a parasite that was brought into Florida from different parts of the world, from Pakistan, from China, from Vietnam, and it's also a good parasite. And the females lay eggs on the body of the Asian citrusillid, and the developing larvae of the parasite consume the body contents, and eventually the adults coming out of mummified nymphs and doing the whole process again. So this parasite is being released here in the state and contributing to the psyllid control as well. We did some studies several years ago to measure the impact of this parasite, and that was based on some very initial releases that were made in 1998 and 2000 in Florida. And at that time, we did not see much contribution of the parasite because those releases were made in limited numbers and for a limited time, and then never followed up. So we did see the parasitism spread across the state, but at very low levels. So at that time, we recommended the augmentation of these parasites, and we brought in new populations from Pakistan, China, and Vietnam. And now the state is producing millions of these parasites and releasing in the environment and giving to the growers for release as well. So this is just to show you that some efforts that went in and the people that were involved, and this process is now happening in the state. So it's highly variable, the parasitism rates. In some locations, we see very high level of parasitism, and obviously it gets impacted by the high use of insecticide as well. So here you see the black and brown bars where there was no use of or less use of insecticides, and we were able to see high level of parasitism even in the range of 40 to 70% at certain times of the years, but where the insecticide sprays were more common, the level of parasitism were very low, either not available or at very low level. So like I said earlier, the dormant winter period, if we can target the psyllids during that period when mainly the adults are available, we can really impact the populations. And then as we move through the year, it's very important to rotate the mode of action so that the pests are not developing resistance. So the sprays are done by aerial or by ground applications. and systemic applications to young trees are done through the branch applications. This is a simple method that I developed back in 2006 and 7. It's called tap sampling method. Very simple, a white laminated sheet on a clipboard, and you can place at random locations under the trees and tap the branches with a short length of PVC pipe. And you can easily count the adults that fall on the sheet. You can also count several other pests and beneficial insect at that time. And we have provided those knowledge and information to the stakeholders in various ways in these informational sheets and documents. So that's another that most of the growers use, and it gives you instant information compared to some of the other methods that were available, such as sticky cards or section sampling. So it's a quick method, and you get the idea of what's happening in the grove at that particular time. We did test the use of sprays during the dormant winter period to impact the overwintering populations. And we observed a huge impact of those populations. If you see these graphs on the left side of your screen showing the populations of ACP in response to just one application of chloropyrifos made in January. And that was the only application made. So that was for the proof of concept, just to see how those effects last into the growing season. And you see the dark bars were the plots that were treated in the dormant sprays. So populations did not really show up until April for several months, 3 to 4 months after the January application. And even still, the numbers were very low compared to the blocks where those sprays were not done in the dormant winter period. So we did not make any application during the growing season. As I said, it was a proof of concept to just show the effectiveness of that method. And then as you look at the beneficial organisms during the growing season, those were equally distributed between the plots that were treated in the dormant period or not treated in the dormant period. Obviously, you need to use the insecticides during the growing season, but you need to be very selective with your choices. So this tactic, it got adopted statewide. There were grower groups in different regions that started to follow that. And just an example, following our studies, here, the first bar shows you the pre-treatment counts of the Asian citrusillate and the post-treatment counts about 5 to 6 months after. You don't see much increase. Same thing for 2010 dormant period and later on in the growing season. Not much increase after the dormant spray applications because you heavily suppressed those populations. We only had the option to keep an untreated control in 2009. And you can see here, there was about 15-fold increase in the psyllid populations if those dormant sprays were not implemented. So these data were collected by Division of Plant Industry inspectors and provided to us. We have also tested a wide range of insecticides that were already developed or developed during the process as we started managing the Asian citrusillate. And most of those provide a very good control of the psyllid, lasting from 2 to 3 weeks and even up to a month or more for the most cases. But again, it's highly variable how those applications were made and the method of applications and all that. But we do have several mode of action that are available to the growers. They can rotate those mode of actions to avoid the concern of pest resistance. Some of the things that we know we need to be aware of as we manage the psyllids, and one of that is that biological control is significant. So we don't want to be taking the actions that can impact biological control significantly. And if we implement the dormant sprays of pyrethroids and organophosphates, we can significantly lower the populations before the season starts. And then there is evidence of resistance. So that's another point of concern that we need to be very careful with our choices for chemical control. So for all those reasons, we also started some programs a couple of years ago to test different organic programs in comparison with the conventional program, so that we can find other tools that can be integrated into the system. So we had a program with organic insecticides only, or organic insecticides with soap or oil, a 15-acre block study for 5 years. And then we had also untreated control, and we were releasing the parasite Tamaroxia radiata in all program. So we're monitoring the populations. We are making these applications based on a treatment threshold, which I'll explain on the next slide. So these were how those programs were developed. Like I said, there are basically two seasons, the dormant winter period, and then you have the growing season. So we tested an organic insecticide monthly applications in these programs, November, December, and January. In one program, only organic was applied. Other, it was mixed with the insecticidal soap, which is emptied. And in the third program, it was mixed with the horticultural mineral oil. It's named 435 oil. So 3 applications, November, December, and January, whereas in the conventional program, just 1 application made in the month of January. As we moved into the growing season, we used a treatment threshold of 0.1 adults per tap sample. So when we saw those numbers, we made the spray applications in those programs, basically organic products only, organic products rotated with emptied, organic products rotated with horticultural mineral oil, and rotation of synthetic products. And as I said earlier, we were making the releases of the parasite Tamaroxia radiata in all those programs. So this is just a summary table of what I said on the previous slide, kind of showing you the number of applications that went in the organic programs, number of applications that went into the conventional program. It's important to note that in the organic programs, I have combined these 3 programs here, but in the 2 programs where we were using the organic insecticide with the soap or oil, actual organic insecticide applications were reduced in these numbers to 50%, because half of those applications were either a soap application or an oil application. And these are the insecticides that were used in those programs. Just to show you the impact of the dormant spray applications, these arrows indicating when the insecticide sprays were made, 3 sprays in the organic programs and only 1 in the conventional. In this year, we made another spray in the conventional program early in the year because population levels were high, and we were worried that not making an application will result in the spread of the salads to the other programs as well. But overall, the organic programs with the soap or oil provided significant % reduction in ACP populations. And also the conventional insecticide Danitol did the same thing. And these level of reductions start to go down as we proceed toward the growing season, because the trees start to produce the new growth and the low level of psyllids start to reproduce, and then the numbers start to increase. So from this point on, there was need of spray applications. Again, basically same setup as on the previous slide, just another season here, and the same levels of reductions that we observed. And again, the need for making the spray application starting spring or a little after. So this shows you the impact of those programs, overall impact for 3 years average, most reduction with the conventional program, followed by the organic program with oil or soap, and then the program of organic insecticides only. Significant effect, but not good as compared to the program with oil and soap. And another year, similar impact that I mentioned earlier. These differences in a conventional program with the 2 of these programs, organic insecticide with oil or soap, were not always that great. So, which is a good thing that we have some other tools that can provide somewhat similar level of reduction. The populations of beneficial organisms, good number of spiders in all programs, same for the lacewings. Lady beetle numbers were significantly reduced in all the programs, and that is the general trend that we see nowadays because of the high use of insecticide that followed the management of this insect significantly impacted the important predators that were effective against this pest. On this slide, you see the parasitism levels of the parasite, the Asian citrus psyllid in different programs. So the blue bar is the untreated, and then the green bar is combined parasitism in the organic programs, and then the red is the conventional program. So we definitely see that the parasite was able to stabilize and contribute to good levels in the organic programs. But in the conventional program, the level of parasitism was very low. And it's not that nymphs were not available. We had the nymphal samples from those programs, but the level of parasitism was not that great. We have also been doing some other research, trying to see if we can establish these parasites more effectively, and basically covering these trees and these large tents, trying to increase the psyllid numbers, release the parasites, and then allowing the parasites to escape from the screens of these cages, but not the Asian citrus psyllid. So, and we have seen that with these type of efforts, we can see that within short period of time, when the parasite starts to emerge from those cages in the field, we observe good level of parasitism in the neighboring trees and within the block, as you see here, level up to 40%, and in some cases we also found 60 to 80% parasitism when we were conducting that research. So we also went ahead and looked at the yield in those different programs, those organic and conventional programs. And you see the data here. Interestingly, one of the organic program in rotation with 435 oil, similar yield compared to a conventional program, although the other organic program with soap was not that great. Actually, the yield went down, and our hypothesis there was that it was the product that got sprayed when the trees were blooming, and that probably affected the tree in some ways, and that's why yield was lower. But the organic programs by themselves were not that great and not different from untreated control. But this was a surprise to us, and we were careful the next season, and you will see on the next slide that it started to improve. As you see here, again, the organic program in rotation with 435 oil, still maintaining the yield while the conventional program even went down. The program with the soap that I mentioned earlier improved. And as I said, it improved further as we moved into those programs. But basically, at least one organic program with organic insecticides in combination with 435 oil did show that there is some hope and tools that can be used toward integrated pest management. For the young trees, we have again tested several products, different neonicotinoid products, as well as some other experimental that were provided to us and needed test. And basically, it was the neonicotinoid insecticides that provided significant production in those programs. We had a new product, a different mode of action, centroniliprole, that is also effective, but the cost for that product is high. So the growers are not using that product to the levels where it is needed. And the issue with the neonicotinoids now that we see is the resistance in the psyllid to these neonicotinoid insecticides, because those were used for several years. And that was the only class of insecticides that was used. And now we have a concern and actually a serious issue of psyllid resistance to that class of insecticide. So we are also trying other ways and means for protecting the young trees. And one of them is that growing those young trees on ultraviolet reflective mulches so that the psyllids can be deterred from colonizing those trees and reduce the HLB incidence. And that helps with the irrigation and fertigation and use of the herbicides as well. And the trees on the mulch get rapid growth, and studies have shown early yield and improved yield. So large-scale trials are still in progress, and we are testing for more knowledge in this system, not only on the Asian citrus psyllid, but with respect to other pests as well. And I have a PhD student, and part of his thesis is some work with this system. When the mulches were tried in one of the previous studies, there was significant impact. The red bars here, you see the yield in those blocks, and also the blue bar is the one, sorry, the blue bar is the one where the mulch was used, and the red is the one where insecticides were used. And you see also providing almost similar yield. But when these two methods were combined, you see here that the impact was even greater. And again, it comes to the point of integrating different methods of pest control because any single method is not enough to deal with this vector pathogen complex. So the spread of the disease and vector continues to be a huge concern. And that's why now some of the growers, especially for the fresh fruit, they are moving to these citrus under protective screen structure. And it's the studies have shown that we can produce citrus in these protective structures without the Asian citrus psyllid and Huanglong Bing disease. And then these are called individual protective covers, mini cups. Obviously, these type of structure, the one on the top, is more useful for small acreage, maybe for the fresh fruit markets, whereas the large acreage, like in Florida that we have, it's not so far feasible to come up with these type of methods, but at least to protect the young trees, similar, and we call them individual protective covers or mini cups. So these these bags of mesh can be placed on those trees for initial few years of growth, so that they are protected from the vector and the pathogen, and can be taken to the stage where they are more strong and able to get into the production phase. I mentioned earlier that citrus is colonized by a wide range of pests, and we want always to convey that to our stakeholders, and we do that from time to time. And also try to come up with the methods like you see here, and we translate that information for growers in the industry articles and other sources. So basically, hello. So basically, there are products that they can choose at different times of the year, and they can choose the products that can target multiple pests, so that they can reduce the incidence of the this vector as well as the other pests, and use less insecticides. These are some of the predators and parasites that are in Florida system, outdoor systems, and important to conserve and augment, although they were significantly impacted by the use of insecticides over the years. And then my lab is also looking at some of the commercial predators that were never used in the system. So we are testing them against the Asian citrus psyllid and see how effective they are. The grower started releasing lady beetle species without consent with us or the other scientists, but they did not see the establishment of this species in the groves. We did test it against the psyllids and aphids, and it was very effective under the controlled conditions when we tested, and developing and reproducing on those diets, but did not really establish in the citrus groves. Then we have seen it in other systems such as vegetables, where it was very useful against some of the mite pests. And we have also tested another species of lady beetle and lacewing against the Asian citrus psyllid under semi-field condition, and found them effective. One of the predatory mites, also very effective, where the mites were released on the psyllid infested plants, and the psyllid emergence was reduced by 80%. We are conducting some more large-scale studies now. We are trying those mites in the fields and see if we can get them established and test their effects. And overall, as you saw, we observed significant impact of biological control if there was less intervention of insecticides, and organic insecticides with oil provided control and yield and indicate potential use in all citrus. Conventional growers can have the option of diversifying their programs by including organic insecticides, and the COPs and IPCs provide significant control of ACP and HLB. Successful integrated area-wide citrus pest management programs require use of all available tools and monitoring for existing and new threats. And we are evaluating based on this information. Right now, we have large-scale studies where we are evaluating different integrated pest management programs and evaluating psyllid management in open and protected systems. With that, I would like to thank the funding sources, state, federal, private industry, help from the growers for conducting all the applied work, Division of Plant Industries, my postdocs, students, biological scientists, and research assistants. And thank you very much for your patience.
Thank you, Dr. Jawad. Any question? أي سؤال يا جماعة؟ دكتور أمنية.
Can I stop sharing?
دكتور أمنية كنت عايز تسألي؟ She want to ask about what effect of weather factor on the population of insects.
What effect the weather factors on the relationship with
Weather factors on population of insects.
Okay, so basically, like I said, we have a dormant or a winter period between November and February, where temperatures are low, and also we don't get much rain, and the trees are not producing the new growth. So that is the time when the most pest populations are low, including the citrus. And as we see the increase in tree growth, start new growth in spring, in February, March, and onward, that provide the chance for the pest populations to increase. And it continues for several months until fall. And with rains, shown that we do see some effect, some dislodging of the insects or the nymphs, but not huge. And also the high temperatures and humidity really, we do see the psyllid populations almost all those months, depending upon what kind of management is going. And I had a student who looked at different temperatures and their effect on the ACP populations. And previous work showed that 28 is the optimal temperature for these insects, but we did see that they were doing fine at 33 degrees centigrade as well, reproducing and developing. Although at 35 and above, frequently impacted their populations. And they were able to get, at 35, they were able to get to the 4th and instars, but then most were not developing to the adulthood, or they were deformed if they did.
Okay, thank you. في سؤال بيقول لم تتطرق الحديث عن بعض الآفات الحيوانية مثل القوارض والطيور والقواقع، أم أنه ليس لها أهمية اقتصادية في الموالح؟ Dr. Jawad? You hear me?
Yeah, I can hear you.
Okay, another question.
Okay.
Translate don't will. Okay. Thank you, Dr. Jawad Quresh from United States of America, Florida University. Thank you, Dr. Jawad.
Thank you very much. Thank you.
Thank you. Professor Dr. Mitchell, are you here? Professor Dr. Mitchell, are you here? اوكي. طب واضح الدكتوره لسه ما دخلتش معانا، هنستنى فاضل لها 4 دقايق وتدخل ان شاء الله. Professor Dr. Mitchell, are you here?
بص يا زعيم، احنا عندنا 3.
So, all right. That's better.
بسم الله الرحمن الرحيم. معنا بروفيسور دكتور ميتشل في الولايات المتحدة الأمريكية جامعة ولاية كانساس. هيكلمنا النهارده عن التكنولوجيا الناشئة في المكافحة البيولوجية للآفات في المحاصيل الحقلية. اتفضل يا دكتور. You can start.
Oh, thank you.
Okay.
All right. Let me see here. Just let me get this going here. Okay. Today I'm going to talk about some of the new ways we can potentially integrate more biological controls into our pest management programs using some modern technologies in some cases. So some of these approaches are proving actually to be more effective than chemical pesticides, and even in large-scale monocultures of field crops, such as we grow here in Kansas. There are success stories from many different cropping systems that can provide us with some lessons, or at least give us some idea about what can be achieved. So the goal is profitable agriculture with reduced reliance on synthetic and costly chemical inputs that damage biodiversity, the environment, and human health. Okay, how's this going to work? So let's see here. As you all know by now, biological control is the use of beneficial organisms to suppress the population of other organisms we generally refer to as pests. Technically, there are three kinds of biological control, but we can ignore introductions as this is the domain of regulatory agencies. I've always emphasized the importance of conservation of plant and insect diversity within and around cropping systems to help farmers benefit from natural or ecological pest control. This is the foundation on which all IPM programs should be built. So we'll start with some general approaches farmers can use to improve conservation biological control. Many field crops have low unit value, so those farmers can't afford the expensive biological augmentation tactics used, for example, in greenhouse vegetable production. For many years, I've discouraged the idea that augmentation of natural enemies can be a viable approach in open field applications, especially considering that the organisms commercially available were generally not appropriate for these uses. But that might be about to change, given recent advances in drone technologies that enable augmentation to be economic on larger scales in outdoor settings, and the commercial mass production of a much wider range of beneficial species. But we should always remember that you can't just sell live biological control agents without providing an education package on how to deploy them properly. So it's never going to be as easy as selling pesticides. When we think about integrated pest management as we did in the 20th century, it was all about how can we get some beneficial species to survive in our crop so we can use less insecticides. The best we could do was come up with monitoring programs and economic thresholds, promote more selective insecticides, and try and limit spraying to the extent possible. But we are now moving beyond that to the integration of biological control with other technologies like plant resistance traits and using combinations of natural enemies to control multiple pests simultaneously. However, this will require a shift away from the reactive mindset, waiting until we see a problem to respond, and toward a more proactive or even preemptive approaches. And recall, that is exactly what we seek to discourage when it comes to insecticide use. So it will require a different pest management philosophy. The idea that we need to maintain a healthy community of arthropods both in and around the crop. Natural or conservation biological control is going to remain the foundation of sustainable pest management in our cropping systems for the foreseeable future. We grow intrinsically vulnerable plants in vast monocultures that make them even more vulnerable to pest attack because of the locally reduced plant and insect diversity. So we need to recognize that agriculture creates pests. The more acreage in one crop, the stronger the selection on insect herbivores to start attacking it, and the more pests it will accumulate over time. We've become far too dependent on expensive pesticides to solve our insect problems, and reducing input costs should be front of mind for every farmer in this challenging economic environment. So consider for a moment every profitable crop you have ever raised as a farmer without spraying an insecticide. Quite a few, probably. Well, that was a crop in which beneficial arthropods, working for free, prevented any pests from exceeding economic thresholds. So why can't this be the case for every crop we raise? The problem is that we often fail to recognize the various ways in which we actively disrupt natural biological control with our agricultural activities, as much as we frequently fail to appreciate how often it benefits us. We need to appreciate the connectivity that links all our different cropping systems. No field exists as an island. In Kansas, for example, we raise huge numbers of beneficial insects inadvertently in our winter wheat crop. And these then disperse into the landscape and they help us with pest control on summer crops. The value of these pest control services is worth some small on-farm investments in conservation, like, for example, including insectary plants in the cover crop mixes we might plant to reduce reliance on herbicides during fallow periods. Our transition away from chemically dependent agriculture requires changing our mindset about what agriculture should look like. We need to move toward production systems more in harmony with nature, even if we only make one small step at a time. To this end, practitioners of conservation biological control seek various ways to renaturalize agroecosystems, bring them back closer to nature, whether by providing floral resources to attract beneficial species, as in the case of intercropping sweet alyssum with lettuce, to enhance aphid control by syrphid flies, or by retaining unmowed borders around cereal fields to enhance the overwintering survival of ground beetles, as they do in Europe. But if you're going to recruit natural enemies into your field, remember they have to come from somewhere, whether it's an earlier crop in an adjacent field or a natural area nearby. We can't just have thousands of acres of corn and nothing else. We really need a mosaic of different habitats in the landscape, and these can be a variety of crops as well as wild areas. For example, recently, the entire US experienced a huge outbreak of sugarcane aphid on sorghum, pretty much everywhere sorghum was grown. On the high plains, it is now generally under natural control after 3 or 4 years. However, it continues to require chemical control on the southern East Coast. Now, why would that be? Well, on the East Coast, they just don't have sufficient numbers of predators colonizing their summer crops. And why do we have so many on the high plains? Because sorghum is preceded every year by millions of acres of winter wheat. And this is what serves as a breeding ground for all our aphid natural enemies that then move into our summer crops in large numbers. This is not the case on the East Coast, which is why they don't have natural control of sugarcane aphid and sorghum like we do here on the high plains. Oops. virtually all arthropod natural enemies, be they predator or parasitoid, insect or mite, rely on floral resources at some point in their life cycle. This is even true for a lot of spiders. Pollen is a good source of protein and lipids, and nectar is a source of energy to fuel flight and can be converted into lipids, which can then extend the lifespan of adult insects. Many predators, such as lady beetles and lacewings, rely on these resources to survive periods of prey scarcity when they are forced into reproductive diapause. Floral resources have very few, if any, substitutes. And yet there is an agricultural tendency to view all wild flowering plants as weeds. In the US, for example, there's a cultural preference for mowed grass along the roadsides in marginal areas. And these grasses do not provide floral resources for beneficial species. So it's a cultural thing. We have to change people's expectations of seeing the whole landscape look like a golf course everywhere we go. Now, we have this concept of insectary plants, or plants that benefit insects in general, the good and the bad. Some of these flowers, such as marigolds, may also indirectly or directly repel pests because of their chemistry. But apart from directly nourishing beneficial species, most flowers will be indirectly repellent to pests as well. Why? Simply by virtue of hosting and feeding their natural enemies. This effect is often referred to as the ecology of fear. Any evidence of predators in the area forces herbivores to be vigilant, so they spend less time feeding and more time worrying about not getting eaten. Now, even if there are some juicy crop plants available to eat, if neighboring flowers are swarming with predators, herbivores may seek to lay their eggs elsewhere because of the risk. As a farmer, the challenge is to find plant species that will thrive under local conditions on your local farm. We know that some classical insectary plants, for example, do not work in our environment here in Kansas, or at least they cannot compete with other species such as oats or black rye that we must include in cover crop mixes for purposes of weed suppression. So these include sweet alyssum, coriander, and buckwheat. So we can't use those. Other areas with better rainfall, perhaps you can. That said, we've had reasonably good results with mustards, clovers, phacelia, which is commonly called scorpion flower, and various asteraceae that thrive naturally on the high plains, including sunflower. Others such as fennel, dill, and marigold remain untested in our environment. So, as I like to say, biological control is not rocket science. It's connect the dots. And there are only 3 dots. We know that biological control requires arthropod diversity, and that arthropod diversity requires plant diversity. But unfortunately, our current agricultural practices pose many threats to natural pest control. And we need to start by considering the soil environment, which actually harbors a greater diversity of life than we see above the ground, even if most of it is microscopic. All agriculture starts in the soil, and healthy soil is alive with biodiversity that has many implications for the health of the plant and its ability to resist pests and diseases. Soil mites, bacteria, fungi, mycorrhizae, all manner of microorganisms have symbiotic relationship with plant roots, help them extract nutrients from the soil, and yes, even help to defend them against above ground pests. So if you till or disturb your soil regularly, you are damaging your soil ecosystem. No-till is always preferable. If you're applying regularly synthetic nitrogen fertilizers, well, maybe that's a system you're locked into currently, but be aware that you're impeding the growth of nitrovying bacteria that would otherwise help you recycle the nitrogen naturally from your crop residues. Which brings us to the seed treatments with systemic insecticides that are currently one of the biggest threats out there to healthy soil and to sustainable agriculture in general. We really have no idea of the range of beneficial soil organisms we are negatively impacting with these neonicotinoid seed treatments. So let's start with the fact that insecticidal seed treatments are not compatible with integrated pest management because there is no justification of their need before application. They have also become another added input cost that the farmer is no longer able to avoid, at least here in Kansas, as so many seed companies are treating all of the seed. You no longer have a choice to buy untreated seed. Imagine what happened to the free market. So these companies are trying to take us back to the 1950s and the prophylactic use of insecticides. This is what integrated pest management was supposed to save us from. Now, sure, you can get some marginal yield benefits from seed treatments, mainly in tropical and subtropical regions where insects are active year-round. But even these supposed benefits do not take into account their many indirect costs. So, all of these neonicotinoids have many different negative impacts on beneficial insects, and these can be both direct and indirect. There are so many different routes of poisoning. For example, these insectary plants I just showed you can become contaminated with the insecticide simply by growing next to a crop plant where the seed, which grew from treated seed. And of course, when they're contaminated, their nectar and their pollen becomes contaminated. And instead of doing something good for the environment, you're poisoning the beneficial insects. And even if they work well and are effective, what does this mean? It means you have zero herbivores in the crop. Even at non-economic levels, it's a scorched earth policy. You start with nobody home. So these pest-free fields result in exclusion of natural enemies by virtue of prey deprivation. There is nothing to eat. So when these chemicals wear off, you have a vacuum which is quickly filled by invasive pests and no natural enemies there to help you. Even more concerned, these neonicotinoids are proving quite mobile within groundwater and highly toxic to aquatic invertebrates and implicated in the large-scale decline of arthropod diversity overall. So think about it. These arthropods are critical food resources for vertebrate species that we value for hunting and fishing. Most sport fish rely largely on aquatic larval arthropods as food, and juvenile game birds eat insects almost exclusively during their first few months of life. So now we're going to switch gears and look a bit at augmentation of biological control agents, okay? And historically, of course, when we think about augmentation and where it has been successful, we tend to think about greenhouse production because we have more of a closed system. So you have some containment of those released organisms, you have some control of the physical conditions, you can manipulate those in favor of your preferred beneficial species. And usually you're dealing with a high value crop like tomatoes or peppers, and you can justify greater pest control expenditures. You also have higher risks of using insecticides in an indoor environment. And there are many consumers who are willing to pay a premium if you can certify your vegetables as being grown without pesticides. But more recently, augmentation has become feasible in a wider range of settings. Not only are a wider range of biological control agents now commercially available, these also now include some generalist predators available for a wider range of applications. So we have this possibility of controlling multiple pests with one predator. And we also have the possibility of synergistic combinations of different biological control agents. For example, using a parasitoid and a predator together. And finally, we have these new technologies, these unmanned aerial vehicles, which are promise far more economic means of distributing released organisms over larger areas in what we're now referring to as precision biological control. So let's start out with some caveats in applying augmentation biological control. We need to recognize not all beneficial species are well suited for mass rearing and release. You can have quality control issues, what I call domestication phenomena. So natural behaviors may be lost in these laboratory colonies. The foraging abilities of these insects can be compromised. It's really hard to rear insects continuously in a laboratory without having some of these domestication phenomena. It's almost inevitable because you're relaxing all the selection that would normally occur in the natural environment. Now, once you have a product developed and you're ready to sell, of course, you've invested a lot in the technology of mass producing this insect. There's always going to be an incentive to expand markets. And that's a concern because it raises because this is a for-profit industry. Augmentation is the business of biological control. Someone's trying to make money rearing and selling these insects. And so there's this risk that in the rush to expand markets for an insect that they're going to be, it's going to be promoted in some inappropriate applications. Now, open field applications of augmentation have always been much more difficult because the behavioral component of the natural enemy becomes far more important and is very often compromised in these mass reared insects. It's much more important in the field. Their complex behaviors and foraging habits and learning abilities, responses to plants, et cetera, are all more important to foraging success in the field. Whereas in a greenhouse, you let them go and put them right on the plants. It's a smaller scale environment. It's not such a problem. And then we have this issue that mass reared insects may lack key adaptations to local conditions. So even if I have one species that occurs over a wide range, at one end of the range, its environmental and behavioral adaptations could be very different from the other end of its range. And yet we tend to treat species as this uniform entity, which it isn't. So there's a lot of regional variation in behavior and biology among populations of the same species that these mass reared insects almost never match. So it's always best to mass rear insects. On the one hand, return new genetic material to the population from the wild as frequently as you can. And number two, work with a population that is locally adapted to the environment where it's going to be used. So let us start with an example of augmentation biological control that doesn't work. And this is a product that, in my view, nobody should be using, even in their home gardens. So here in North America, we have a long-standing industry, and it goes back decades. These convergent lady beetles that are sold by mail orders and by many commercial outlets across North America. These are not mass reared in an insectary. They are mass harvested from their overwintering sites in the Sierra Nevada mountains. So they form these aggregations. They're literally shoveled into buckets and put into cold storage and then sold. Now, this makes them a very low-cost commodity with a high profit margin. But is this sustainable for those populations? And more importantly, they don't work. And why don't they work? Well, there's a lot of reason. Of course, everybody likes the idea of releasing ladybugs in their garden. But the problem is these are very old beetles that have overwintered. They're strongly dispersive. When you warm them up and they break hibernation, they want to fly away. And because they're old, they have very little reproductive potential. And that's what you really need. You want those beetles to go and lay eggs on your plants because it's the larvae that are going to swarm the plants and eliminate all the aphids. To make matters worse, they have different levels of parasitism or disease. Up to 20% can be parasitized. We've sampled these packages. Or they can be diseased. There's different types of fungal diseases that infect lady beetles. And of course, this is a population that's isolated on the west of the Rocky Mountains. And they may have levels of immunity to these parasites and diseases they've evolved with that is not shared by populations where they're going to be released. And they're not adapted to survive in these other areas where they're released. They're adapted to Central California. And so, but they will interbreed with local populations because hippodamia occurs all across North America. So this isn't good. This causes genetic admixture of maladaptive genes in the local populations. And I mentioned the possible impacts on source populations. But at the end of the day, they fly away. The homeowner comes back the next day and they don't see any beetles. And they say, well, that's it, biological control doesn't work. More generally is their assumption. So it gives biological control a bad name. So if that's an example of something that doesn't work and why, let's review some of the properties of beneficial species that do suit them for use in augmentation, as well as some that do not. And so generally, these are on the left here, you have a list of organisms which have generally been successfully used in a lot of different, mostly greenhouse environments, but of course, predatory mites, nematodes, various pathogens, fungi and nuclear polyhedrosis viruses. Egg parasitoids, I'm going to cover in more depth because they are one of the first examples of augmentation biological control going back 100 years. Certain larval parasitoids are well suited, and some predatory midges. So then generally the properties we're looking for are something that we can mass rear very cheaply, so we can afford to throw them away if we don't sell them. Something with few or no inbreeding problems. This is where these Hymenopterous parasitoids come in. They are haplodiploid, so there are no lethal recessive alleles hiding in the population. They're eliminated every time they're in the males because the males only have one copy. So you don't have inbreeding depression in those populations in the lab, even though they've been bred there for years. And the practicality of releasing and distributing them is another factor. And so that you want that to easy to get them out there. And finally, you want something that's weakly dispersive. It has to be able to disperse somewhat. You don't want it to, you want it to work where you release it. So we can look on the other side and you'll see a lot of our good generalist predators on this side, like the lady beetles and the lacewings and the hoverflies. And they're not as well suited. I'm going to say you can't rear them and release them. You can. One of the problems is they're much more expensive to rear. So all of these species here really have a problem with larval cannibalism. You have to isolate them in the larval stage because they're very aggressive and competitive. So they kill each other. You're rearing these chrysopods. You have to keep each one isolated in its own cell. And you can't have inbreeding issues because these are not haplodiploid like the parasitoids, Hymenopterous parasitoid. They're not so easy to distribute in the environment as easily either, if you think about it. First, you have to decide on what life stage to release. And of course, eggs are susceptible to predation or even cannibalism. And adults are the most robust stage, but of course, they're strongly dispersive. They can fly. And so there's a lot more issues. I won't say you can't augment these predatory species, but there's more issues associated with, some of which we're starting to solve. So let's look then at these egg parasitoids. Where is that? Okay. So trichogramma are the very first parasitoids ever mass reared for release in agriculture, almost 100 years ago. And they've been very effective in many different contexts. They're easy to mass produce, and you can use a lot of different factitious hosts, not necessarily the one that you're going to be targeting with it. Often they can parasitize a range of hosts as well. You don't have problems with inbreeding because they're haplodiploid, and they attack a wide range of moth and heteropterous pests in the egg stage, which is good because then if you can kill it in the egg stage, you don't have any feeding damage, of course, on the plant at all. So here's one development. I'm going to talk about another one technological development for distributing these parasitoids. See, there was a lot of research into using egg parasitoids to control corn borers in corn. But the advent of BT corn ended that. But now we have a lot of other moth species causing losses in our grain crops that are not controlled by BT. And others are evolving resistance to BT. So in this case, what they've done is they packaged these parasitized host eggs. So the parasitoids are in the pupal stage in there. And they package some with unparasitized eggs, but also in different stages of parasitism. So what you have are up to 6 different waves of wasp emergence. And even if some of the first wasps to emerge will go around and attack the unparasitized host eggs that are in there, have another generation in that little biodegradable capsule before they leave. And those unparasitized host eggs, incidentally, are irradiated so they won't hatch if they're not parasitized. And so you have this way of delivering those, releasing these things in a sort of temporal presence within the crop over some time, up to 2 weeks. But recall, these are tiny wasps with very low dispersal capacity. And that's an issue. So what do we used to do? In the old days, the convention was to coat pieces of cardboard, like you see here on the right, with honey, and then glue the parasitized eggs to the card hanging on the plant. So you have probably 2,000-3,000 parasitoids right here. Now, for many years, the Chinese were using aircraft to disperse billions of trichogramma over agricultural fields on very large scales. The idea that being that they would get started on non-pest hosts, and then as the pest built in the crop, they would migrate in from the marginal areas, and it works. But you think about how wasteful that is, using an aircraft and a pilot, when your payload weighs only a few grams. And the alternative, hanging cards, well, again, you know, how many cards can you hang? How far apart do they have to be? I've shown here these circles sort of represent that 10-foot radius, which you're going to get dispersal from those cards. And so you have a lot of area that isn't really treated. If you're only going to hang a card, for example, every 100 feet in an orchard. So you have problems, you know, really distributing them effectively in the environment. So here's a new technology that's come up to deal with that. And we're now in the drone age, and drones are far more economical aerial distribution systems. Now, this company based in Central America developed a flowable formulation of parasitized eggs. And this was the key to effective aerial formation. So the eggs are covered in a proprietary coating. They're very, very tiny, but they look like a fine black sand, and they're all slippery. So you have 20,000 parasitoids in every cubic centimeter. Think about it, that's a sugar cube. And so with one small drone, they can treat 1,000 acres of sugarcane a day at a tiny fraction of the cost of a plane. Plus, once you've saved a GPS program to treat a particular field, the process is completely automated. So what do you do about the issue of temporal persistence? Well, in this case, we're controlling caterpillars boring sugarcane, so we can follow up with releases of larval parasitoids to attack any borers that escaped the egg parasitoids and got into the stalks. Now, the problem here is we're dealing with larger pupae, and they're not coated, and they're vulnerable to predation prior to emergence. And in sugarcane, there's many ants which will eat them. And so the parasitized pupae are coated with an ant repellent. They only need 24 to 48 hours protection for the parasitoids to emerge successfully. It's very cost effective compared to insecticides, and it works. And in terms of the distribution of putting 50,000 wasps per hectare, they've actually, the egg parasitoids, they've actually shown that they can precisely deliver 8 eggs for every square meter. So that's a lot of accuracy, and you get a more uniform distribution across the field here than you have with the cards. And there are many other applications now that are taking advantage of drone technology for augmentation of natural enemies. And it's all comes down to these different designs of the dispensers that we've developed. And most of these are specific for particular biological control agents. So in this case here, this guy, you see these strawberry fields, that's a high value crop in California and Florida. Well, these drones can go up and down the rows and sprinkle evenly predatory mites across these plants, because spider mites are a big problem in strawberry production. Over here on the right, you see here, the parabug system. What you have here these are rice hulls used as a carrier. And in there will be newly hatched predatory larvae like lacewings. And the rice hulls is harborage to keep a complex environment. And mixed into that will be some food eggs, probably a Festia. And so by timing this carefully, when the lacewings are just hatching, they can sprinkle them across the field. And they're using the system to augment predators and parasitoids, for example, in citrus groves. And you can see some of the other different types of dispensers that have been designed for particular biological control agents. So as I've mentioned, there's never been a wider array of mass-reared beneficial organisms available for augmentation strategies, which raises the possibility of applying them in various combinations to increase their efficacy. So the example many of you are probably already familiar with, if you work with copper, you know, the use of an aphid parasitoid in combination with predatory gall midges to control aphids on peppers and tomatoes in greenhouses. Now, there's another interesting example comes from sugarcane production in Colombia. And sugarcane is a large acreage crop in Colombia, and yet they're doing augmentative releases of Cotesia, which is a Hymenopterous ectoparasitoid, very interesting and used against a lot of different Lepidopterous pests. And it's an ectoparasitoid, but it also attacks hosts that are 100 times its size or greater. It's a tiny little wasp. And so what it does is it goes around and parasitizes a bunch of these larvae and injects venom and waits for the venom to take effect because they can be killed by the host. They're so large. But of course, when it does the return trip and looks for all its parasitized hosts, it never really finds them all. So it ends up paralyzing a lot more hosts than it parasitizes. Now we're dealing with a complex of 5 species of Deratrea stem borer in this complex. And tachinid flies have also been successfully mass reared and released against them. In fact, that was the primary control until they had a couple of new species of stem borers show up in the complex, which are much more resistant to parasitism by the tachinid flies. And the tachinids are good because they lay larvae at the entrance holes of the boreholes, and those larvae will quest down the hole and find the stem borer inside. Now, it turns out all those hosts that were stung but not parasitized by Cotesia now become actually more suitable as hosts for those tachinid flies. And so we have one parasitoid actually increasing the suitability of some of these hosts for the other one. Another example comes from control of Helicoverpa armigera in chickpea production, which might be a system close to what you might have in Egypt. In this case, a very well-known braconid parasitoid, which is also gregarious, Heberobracon hebetor, is used in combination with a nuclear polyhedrosis virus, which is highly specific to just that one moth species, Helicoverpa. So what you do is you spray first the formulation of the viral particles to initiate the infection, and 3 days later, you release all your parasitoids. Now, the parasitoids are not affected by the virus, and they can actually develop into diseased hosts, provided the disease does not kill the host before the parasitoid completes development. However, in later stages of the disease, the sluggish behavior of the larvae cues the parasitoid to avoid it. And so actually what you get is synergism between these two biological control agents, because as the parasitoid goes forth and attacks the remaining larvae, it also serves to vector the virus through the field and initiate secondary infections. Similar effect with a predatory soil mite that we've augmented in mushroom production. So in mushroom production, of course, you have, they're grown on manures and in special greenhouses in the dark, and you have problems with these fungus gnats attacking the roots of the mushrooms. Well, the predatory mites are good at eating the fungus gnats, but they don't eat quite enough of them. And so if you combine that with a treatment of the entomopathogenic fungus Metarhizium, does not affect the soil mites at all. And again, the soil mites help to spread the inoculum, the conidia of the fungus throughout the growing medium. And so you get actually more disease and a better epizootic of the fungus in the gnats than you would if you didn't have the soil mites there. And finally, we have an example here of using combination of Beauveria, 2 different fungi, against root-feeding beetles in soybean. And this is an example that comes from Brazil. So the application here is Brazilian soybean fields. So it's a tropical environment. And one of the reasons this works is that we are now finding that many of these pathogenic fungi can grow endophytically within plants. They don't harm the plants, but they can stimulate plant immune responses, which can make the plants more resistant to pests. And of course, they can infect the pests directly that feed on the plant and ingest them. And so these 2 fungi have different properties. Of course, Beauveria there are many strains. Some are more specific than others. Some are very generalist and can infect a wide variety of species. So here in this case, it would be a cicada. But the Metarhizium has different properties. It's a different family of fungi, and it's many isolates that are highly specific to different hosts. But it tends to also have longer persistence under the soil, at least under these tropical conditions in Brazil. And so what they're doing now in Brazil is they're using both of these fungi as seed treatments instead of neonicotinoids for control of root-feeding beetles in soybeans. So we're dealing here with click beetles and scarab beetles, which are known respectively as white grubs and wireworms, and problems, of course, in the soil pests in many crops all around the world, these families of beetles. And what we see is that the Beauveria is actually more effective against the adult stages that will be entering the field first to lay their eggs in the soil. It's fast, it kills more of the adult beetles faster before they'll lay eggs, but of course you don't get all of them. And the Metarhizium that persists longer is actually better against the developing stages that come out of the eggs of the adults that didn't get killed by the Beauveria. So a nice fully biological alternative to using toxic chemicals for protecting the seedlings. So finally, I'm going to look at this here. Let's look at an augmentation strategy that's actually working in Kansas cropping systems right now at a really large scale, because we have fields that are 100 to 200 hectares or more. And so this is the augmentation of these nuclear polyhedrosis viruses to control particular caterpillar pests. And these in this case are highly specific, so they only infect one species of host. In fact, that's where we found it. It's been isolated from that species and nothing else. And so we've developed the technology to produce these viruses in large vats, fermentation vats, in very large quantities, very cheaply. Now, the advantage of the viruses is they don't depend on humidity or moisture for infection, like the fungal diseases do. However, they must be consumed by the caterpillar. And you say, yeah, well, they're not going to kill it right away. But yes, they take 5 to 7 days to kill, but those larvae will stop feeding within about 24 to 48 hours after they're infected. And the nice thing is, once you get this disease seeded into the field, it can self-propagate. So as those larvae die and become pools of ooze, as you see here, they contaminate the feed of other insects that spread them all through the field, leading to secondary infections. So control is actually better when you have a healthy community of insects in the crop, because any insect other than the target caterpillar will act as a vector of the disease without being affected by it. You never need to come back and treat a second time. Plus, we've got problems here because our primary problem, Helicoverpa, is Zea. And, you know, this is we call it corn earworm. It has different names in different crops, but it's been exposed to the same insecticide modes of actions on multiple crops for decades. And so many populations have evolved cross resistance to both organophosphate and pyrethroid insecticides. And so they're really, you know, you can spray these generic materials, people choose them because they're cheap, but they don't work. these insects just laugh it off and keep growing. Now, the commercial virus formulation is called Heligen, and we've had really good results with it the last few years in central Kansas. And it only costs $3 to $5 an acre, plus the application costs. So that's very cost effective, very actually cheaper than most of the insecticides that will work. For example, diamides and spinosad-based insecticides, they still work. There's no resistance against those, but these are newer modes of action, are significantly more expensive. And so this is really not only a biological alternative, it's a cost-effective alternative. And now they've got a product for the fall armyworm, which would be Spodoptera frugiperda. And a combined product is coming soon. So with the mixture of both viruses, you can kill both pests. So because it can be produced, it's not just used in sorghum, which was the crop you just saw it in, but in cotton and soybeans. But you have to apply it early in the crop. So we don't wait until the threshold. It's kind of more of an insurance policy. As soon as you see any small larvae showing up, you want to get it out of there, get it out there. And it's labeled for fumigation, so you can apply it through the overhead pivots, which is very convenient if you have an irrigated field of soybeans like this one down here. And it's compatible with various tank mixes. For example, if you're going to mix it with some fertilizer, but you got to watch the pH. So you want to keep low pH because the virus comes packaged as occlusion bodies. These are the way it survives when it's outside the host. And these acute occlusion bodies are activated when they're ingested by the alkaline gut of the caterpillar. So you don't want to have a high pH tank mix because you'll activate those occlusion bodies prematurely. And so it works well in combination with all other naturally occurring biological control agents. Not only because it doesn't kill non-target species, but because other biological control agents that may be acting here as scavengers on the dying insects serve as vectors to distribute the virus even more widely in the field. So you have some coccinellid beetles here feeding on the diseased and dying larvae, and some ants, and over here a predatory bug. And the interesting thing when we do these tests, and here you see I've worked mostly in the sorghum, what you find that we'll leave a control field adjacent to the one we treated for comparison, and yet we have these disease larvae showing up even in the control field if we don't have it far enough away. That's how much other insects move the inoculum. So in sorghum, we apply Heligen right as soon as the crop reaches flowering. And you can scout sorghum by beating heads into a bucket, but treatment must be applied when the first larvae are still small. You can't wait until a standard threshold we use for chemical application, which is 1 to 2 larvae per head. You'd be too late. So the occlusion bodies, as I mentioned, improve their survival in the environment. And because the disease self-propagates, there is no need for reapplication, even if more eggs are laid in the field by the moths later on. And of course, because no other insects are affected, there's no disruption of biological control agents, and you're not resurgent, you have no resurgence of other pests because you haven't killed any beneficial species. Now, interesting thing here, this is results of some of our work actually in China with the Helicoverpa armigera version of the virus, but you'll notice that the larvae tend to die at the very tops of the plants. And this is because the virus induces a climbing behavior in its host right before it kills it, called tree topping behavior. And so the work we did in China on a very similar system showed that the virus actually upregulates genes in the eyes and nervous system of host larvae, making them more sensitive to light. So the virus hijacks the visual perception of the host and makes it climb to the top of the plant at a time when healthy larvae would normally become photonegative, descend to the soil, and pupate in the soil. So this is beneficial for the virus because it maximizes its dispersal in the environment. It makes the dead cadaver conspicuous to those scavenging insects, and it increases its chance of infecting more hosts, even through passive dispersal like wind and rain splatter. And so here's a little bit of data just to show you something interesting. And you can see the behavior here over time of virus-infected caterpillars in a climbing apparatus. So here in this apparatus is a big glass tube. We've shrouded it in cloth so it's in darkness. And we place a light at the top, caterpillar on this strip of wire mesh. And then we monitor the location of the caterpillars using time-lapse photography. And if you look, the control caterpillars, they'll do some climbing early. So this is now hours after inoculation with the virus. But you'll see with progression that over time, all these healthy control larvae, in fact, they do a bit of climbing. At the end of the experiment, they're all at the base of the apparatus. In contrast, the ones that have been inoculated do a lot more climbing later. And in fact, you see most of this climbing occurs during daylight hours rather than at night because they're responding to light. And instead of becoming photo negative at the end, they end up dying at this heights on the plant up here. And finally, we did a transcriptome analysis of the brain tissues. Actually, we did it again with the stomata, the actual larval eyes of the caterpillar. And we found all these different 361 differentially expressed genes, 231 upregulated and 130 downregulated. And 5 of these genes that we focused on were ones associated with photoreception or phototransduction. In other words, transmission of the photoreceptive signal to the insect's ganglia or brain. So these are 3 of these were opsin genes, which are actually light sensitive pigments in the retina of the eye, sensitive to particular wavelengths of light. And then we have this regulatory gene associated with options. And this other one here, this retinal degeneration gene C, was downregulated. And actually, this gene is putatively responsible for the normal deactivation of the retina as the larvae approach pupation, thus reducing responsiveness to light and driving healthy larvae down the plant to pupate in the soil. And so that the virus actually deactivated that gene to prevent that behavior. So really interesting. And I would have to say, I think the future is promising. While we remain reliant on chemical and pesticides in many cases, newer biorational materials hold promise. And as we get more of them, they will become cheaper. Halogen and phologen are not the only new insect pathogens to get registered in the past 2 years, but they're not going to be the last. And the more biocontrol we can rely on in our cropping systems, the lower our input costs will be. And as we all know, input costs are really eating into the bulk of farm revenue right now. And consider the benefits to farmer health and the environment as a bonus. So the better we can renaturalize our agroecosystems, the fewer pest problems we're going to have in the first place, because we'll have a healthy, robust community of arthropods there, which will suppress the tendency of any one species to become too abundant. And renaturalization can mean different things, different farms. If you plant cover crops, you can include some flowering species. You can manage field margins, so the unplanted areas, as reservoirs of beneficial insects instead of making them look like golf courses. And finally, the term biodiversity has long been considered an undesirable term in agriculture, a concern for environmentalists rather than farmers. But my take-home message today is don't fight biodiversity. Find ways to make it work for you. And with that, I will conclude.
Thank you, Dr. Mitchell. Thank you. في أسئلة يا جماعة؟ Any question?
في ناس رافعين ايديهم يا دكتوره، في حوالي 10 أو 8 رافعين ايديهم.
طب اوكي، خليهم يتكلموا.
دكتور رضا عبده. تفضل يا دكتور عبده، تفضل سؤالك.
أعتقد أن هذه جزء حاجة لا تعرفها وتخفيها
دكتور رضا عبده.
وكيف يمكننا تحقيق ذلك على المستوى الإقليمي؟
طيب دكتور أحمد. دكتور أحمد طه، تفضل.
I want to ask you about the spiders. You didn't speak about the spiders in the field. Is there a new applied technique to use spiders in the field?
No. In fact, spiders are very generalists. And of course, you've got a lot of different, you get web builders, you've got ambush spiders, and you've got the roaming sort of wolf spiders. We know a lot about the physical conditions they need. And we consider them that we haven't got, first of all, there are no technologies exist for mass rearing spiders effectively. And they're the kind of thing, again, if you just start managing your agroecosystem in a more holistic way, spiders are something that will come all by themselves. They balloon in on silk when they're little spiderlings. So we have massive dispersal of spiders by themselves from pastures and other areas. So spiders are definitely a component of the system, but we don't manipulate them directly.
Yes, okay, but I use it in treatment, but it was a very limit under greenhouse control. My asking about the unlimited treatment, if we can.
Again, we don't have any technologies developed to produce large numbers of spiders to release them. But again, spiders are something that you shouldn't need to release in the outdoor setting. They come all by themselves.
Yes.
The only thing, what happens, the thing is, another thing is it's difficult to justify their augmentation because they don't feed on just one thing. They feed on anything that they can subdue. And so, it would be difficult to make a recommendation to a grower that would economically justify augmenting spiders. Does that make sense?
Yes, okay, I get it. Thank you.
So some other questions here in the chat I can deal with. The role of plant extracts in controlling insects in America. Well, I don't consider it a kind of biological control, but that really has very limited use in our system. So for example, there are some neem products that will be used, but this is almost entirely confined to organic production where they don't use alternatives. So botanical insecticides are something that may be feasible. I know there's a lot of research on South America, botanicals, but here we don't use them. We don't use them except in organic production. So there's no real push for them. And I have another one here on BT. Where was it? Importance of pathogenic agents, especially BT. What is my opinion? The problem with BT, now I will say there's a lot of new events out there in the private sector. So these are new mutations which have Bacillus thuringiensis toxins, express new toxins, because there's a whole family of them, and it's a very complicated toxin, and new forms of expression in different plant parts. So, there's a lot of research on that, a lot of new BT products coming. As a formulation that we can spray, the problem is it doesn't last long in the environment when it's applied as a dust or a liquid formulation. It's too sensitive to sunlight. So in bright sunlight, I've used it here against caterpillars in Kansas. Well, you have to apply every 3 days. And if it's expressed in the plant, the problem there is we have too much reliance on BT. So all the corn expresses Bt. Now, a lot of the soybean expresses Bt. So what happens when you rely too much on one technology? Pretty soon doesn't work. And we're already seeing certain insects developing resistance to Bt. Corn rootworm's a good example. We thought that Bt would be very effective against corn rootworms. We found the event to express it in the roots. We have 5 or 6 events, different genetic manipulations, but it's not working so well as it did for the corn borer. The corn borer was a huge success. Yes, everybody abandoned study on corn borer. Bt was the answer. But it's not been the same for other pests. And the rootworms, they can feed a little on the root, and then they can feed a little on some weed, and so they can get a sublethal doses. Also, their behavior in the crop is different, and we're getting a lot of resistance in the corn rootworm to these Bt events. So my issue with Bt, it's a great product. It has its niche. It has its situations where it's one of the best things we can use. But we don't want to rely too heavily on one thing. And the problem with the plant incorporated protections, as we call them, when the plant expresses it, it's expressing it constitutively. So all plants have Bt expressed in those parts all the time. So you have continuous selection pressure on the insects to overcome this resistance. So that's the problem with Bt. We're relying on it on too much in too many different crops. I can't quite make sense of this question. So we've got a question. Is there a program that's used to exchange between predators, parasites, and an entomopathogenic fungi with some pesticide? Yeah, well, if you mean replace pesticides with these products, that's the goal. That's what we're trying to do. And in many cases, where one biocontrol agent is insufficient, a combination of the two is actually better than either would be alone. So we can have an additive effect, or we can have a synergistic effect where the two together are more than the sum of the parts. So we are only just beginning to look for these relationships and take advantage of them. So I think there's great potential there to displace pesticide use in many, many contexts, if that's what the question was. If it was regarding combining them with pesticides, no, because that's not the objective. The objective is to get rid of the synthetic chemistries, because they always have too many non-target effects within and outside the crop. Any other questions?
No, thank you, Professor Dr. Mitchell.
You're welcome.
Thank you. Thank you, Doctor. From United States of America, Kansas University. Thank you, Doctor.
Thank you. Bye now.
Bye. دلوقتي مع دكتور لورا. Are you here? Dr. Laura?
Yes.
Yes, welcome.
Thank you.
دكتور لورا من الولايات المتحدة الأمريكية، جامعة بوردو إنديانا بوليس. هتقول النهاردة محاضرة عن الإدارة المتكاملة للآفات في بيئات غير متخصصة لإنتاج المحاصيل. Can you start, please?
Thank you very much. I'm really excited to be here. Thank you for the invitation. I would like to start with just acknowledging all of the funding support and the students, faculty, and staff that have helped achieve the work that I'm going to be sharing with you today. So the majority of my work is funded through various programs sponsored by the United States Department of Agriculture. And I've had a wide variety of graduate and undergraduate students and faculty mentors that have helped me in the past few years as I have just joined the faculty here at Purdue. So I would like to begin with a snapshot of the industry in terms of specialty crops production in the United States, then take you through the work I have done in high tunnels or hoop houses, as you may know them, Controlled environment production in the strictest sense, which is increasingly common in reclaimed warehouses and other repurposed buildings in urban environments. And lastly, talk about some of my newest work with an emerging specialty crop here in the US. In terms of my personal background, I am broadly trained and interested in ecology, plant-insect interactions, and developing solutions for pest management in sustainable agricultural systems. I'm highly motivated by stakeholder needs, and my primary appointment as an extension faculty provides me with the opportunity to engage with farmers on a regular basis. I am working towards developing an integrated program to support sustainable food systems, working with multiple disciplines to factor in growers' preferences, market opportunities, customer needs and demands when developing insect pest management recommendations. So I just want to set the stage for vegetable production in the US. There is the United States Department of Agriculture National Ag Survey and Statistics Center, which can provide a lot of information about acreage and economics. And yearly we have a publication called the American Vegetable Grower publication, which is a sort of a private journal, but they conduct their own survey to report on the state of the industry. And this is really a good reference to understand production strategies and behaviors and get firsthand experiences and insights from particular growers. So here are some things that I would like to highlight from 2019. There were 549 participants. 379 of these are specialty crops growers. And what we can see here, if we look at the top issues sort of challenging their ability to grow and sell their crops, pest issues were ranked as number 2. 50% of growers use biological methods to control pests, and of that, more than half use insects. We have seen a bit of a decline from 2018 to 2019 in respondents of this survey, but this really highlights that beneficial insects are a large sector of pest management in this industry. So, more than half of the growers that are participating in this survey grow under some sort of cover. And the way that we characterize this is in greenhouses. Okay, these are usually equipped with electricity, so they have active heating and cooling structures. Availability. The second one is high tunnels. So this is a less technical covered growing environment. They're often also called hoop houses, and it's just a large rounded frame with plastic stretched over it. It relies on solar radiation capture for heating and ventilation for cooling. So there's often no electricity or supplemental heat for these structures. And lastly, they have a category called low tunnels, which is something just like a sometimes referred to as a caterpillar tunnel or just a cloth stretched over the crop on the ground. And the reason that growers are using these structures is mainly for season extension. So in temperate climates where we only have 3 or 4 months of ideal crop production here in the Midwest of the US, we can capture the sun's radiation under these structures and begin to grow earlier in the spring, extend production and harvest later into the fall, and sometimes even go year round with that. And oftentimes these structures, particularly greenhouses, are being used to produce transplants that will then go out into the field. And so that's an image of a greenhouse. Here's more of the rudimentary high tunnel structure. And then lastly, we have these row covers. And one reason why we see a really large spike in high tunnels in particular is because there's a USDA funded program to help offset the cost for growers in investing in these structures. So we've really seen a gradual and steady increase in the number of high tunnels found throughout specialty crop production, even making their way into urban farms and very small farm settings. And so like most of the protected agriculture, high tunnel production is a valuable season extension tool. However, it does create this unique hybrid of field and greenhouse conditions that's really dependent on the time of year in which you're producing in them. In the shoulder seasons, as we call them, so the early spring, or the late fall or even winter, it's much more of a closed environment like a traditional greenhouse. So very little movement from within the tunnel to the outside surrounding environment. However, during the peak of the growing season, for us, that's typically 3 months in the high summer. That structure is vented as much as possible. And in that situation, you have very free movement in and out of the growing environment. Because these do have an investment cost related to building them, the market is dominated by a few, but really economical crops. And so tomatoes are the #1 crop that we grow in these systems. That's because they come with a very high value at markets, and you can get the crop to market earlier. This is one of our most popular vegetables at farmers markets and direct to consumer sales outlets. But we do see some diversification, and some of the more emerging and popular crops include cucumbers, bell peppers and hot peppers, and lettuce or mixed green varieties. And so why am I so interested in working in high tunnels? Well, the information that's available for growers is very misleading. When I started working in this system, there were essentially no peer-reviewed publications on pest management tailored specifically for the growing environment. And if growers were to look in the extension publications that were available, they would see a lot of recommendations that were pulled directly from greenhouses and sort of put as a space holder in these high tunnel production guides. And so the first thing that we did was really take a comprehensive and comparative look at what are pest dynamics and pest pressures like in high tunnels compared to open field production. So we had a master's student here who grew tomatoes, cucumbers, and broccoli in high tunnels and had an adjacent plot right outside the high tunnels, so in between the space here of the same crops. And we just monitored differences in pest pressures between these two systems. And what we found was a little bit alarming. So actually we saw what we expected, which was the greenhouse pests thriving in this protected environment. So soft-bodied insects like aphids, mites, and whiteflies. But what we also saw was an even higher pressure from lepidopteran pests. So for us, that included tomato fruit worm and a lot of the crucifer caterpillar complex. So diamondback moth, cabbage looper, and PRS rape. So we're actually seeing higher pressure for growers to deal with in high tunnels compared to open fields. And so then I was interested in looking at some pest management strategies, and a natural start was to work with what does work well in greenhouses and how can we translate that to high tunnel systems. So it's very timely that I'm talking after Dr. Mitchell, because I'm going to talk a lot about biological control and natural enemies today. So the first thing we started with was augmentation biocontrol. So like Dr. Mitchell mentioned, it was very successful in greenhouses. And again, that's because we're limiting the dispersal capabilities of this insect by trapping them in the environment. This success in greenhouses has led to a thriving market for commercially available predators. And many of these insects feed on pests, but also feed on pollen and nectar. And some of them have been shown to be attracted to HIP-Vs, which are herbivore-induced plant volatiles. So this is the smell that a plant gives off when it's being chewed on or attacked by an herbivore. So an olfactory response that some of these natural enemies have learned to identify to locate prey. But what we really need to consider about the uniquenesses of high tunnels is this dispersal capability. So depending on the time of year and the food availability, we expect dispersal to occur, much like it does in open field application. So the objective of this work was to examine the efficacy and retention of predatory insects in high tunnel tomato and cucumber production. And so we set up a system here at our research farm where we looked at conventional production. So this is a tunnel managed with open ventilation to provide the most optimal temperature that we can achieve for tomato and cucumber production. So the tunnel is split right down the middle, 2 rows of tomato on the left, 2 rows of cucumber on the right. In the first treatment that we imposed, we covered all of the openings of the high tunnel with an insect screening material. So this was so it's LS EcoNet 4045, so 0.04 by 0.045 centimeter holes. So it was very, very small. And then in the last situation, we diversified the crop and we incorporated the commercially available predalure. So that's methyl salicylate, that herbivore-induced plant volatile, that smell that plants give off when they're being fed upon by an herbivore. So you can buy this in a little lure and hang it inside of outside in your crop or inside the high tunnel in this case. So we hung 2 lures in this system, and then we have 1 row of tomato and 1 row of cucumber down the inside of the tunnel. But on the outside edges this time, we replaced that vegetable crop with a flowering crop. So here this is a flower that is used as a bouquet filler. And so it's called Gomphrena is the scientific name. And so we placed that on the left-hand side of the tunnel, and we used zinnia on the right-hand side of the tunnel here in this picture. And so we chose those 2 flowers in particular because if we want to convince a grower to replace some of the fruit or vegetable crop in this space, it needs to be something that they can also market and sell. And so these 2 flowers are varieties that are often used in fresh cut floral bouquets. So they could also harvest these flowers and sell them at market. And then we looked at 4 different commercially available predators. So quickly, the green lacewing, the convergent lady beetle, the minute pirate bug, and the spined soldier bug. So this is Chrysopocarne, Hippodamia convergens, Aureus insidiosus, and Podicis maculaventris. And so what we did was we identified sentinel prey plants where we infested them with either aphids or small caterpillars in the morning in the high tunnel, and we flagged those plants. Then we came out at night, we closed the sides of the tunnel, and we released natural enemies. We only did 1 natural enemy at a time, so there was no complementarity between these. It was just 1 sole predator. And then we would come back at various intervals after release and use a DVAC sampler to suck all the insects off the plant and to recapture the natural enemies and see what our retention rates were like. And this was very easy at first when the plants were small, but as they matured into the growing season, we were often collecting over our heads on the top of the plants. And so the data I'm going to show you here is our retention data. So on the y-axis here, we have the proportion recaptured. And on the x-axis here, we have each of the predators that we released. And this is looking at that sample 6 days post-release, when we destructively sampled and pulled all of the insects out of the tunnel. And we have our 3 different treatments. Conventional is in black. The flower and herbivore-induced plant volatile treatment is in gray. And the screen treatment is in hatch marks. And what I want to draw your attention to here are 2 natural enemies that we saw the most promise with in this experiment. And so while our retention rates are quite low, this is close to what you see in some successful field releases, so not too surprising. And the most exciting response was of aureus to that diversified floral and lure treatment. So we saw extremely high, in comparison to the others, recapture rates when we diversified that crop, provide that floral resource and the lures. The other insect that we saw promise with was the green lacewings. And so again, here we were releasing adults for ease of recapture, but really the larvae are what's going to do the work in terms of pest suppression. So we did notice a lot of oviposition or egg laying from these insects. And when we started to collect this information, we noticed some preference in terms of where that insect was laying its eggs on the plant. And that had some implications for growers. And so if we look here and we divide the plant vertically into 4 sections, where number 1 is the top of the plant where the newest tissue is and the growing point is. And moving down 2, 3, and 4 is the base or the bottom of the plant where the old leaves are. We see that on tomato and cucumber, a preference for lacewings to lay their eggs, on the bottom portion of that plant. And so they're really utilizing lower, and this could be a microclimate issue where it's cooler down here than it is up towards the top where the plastic is, or it could be related to the actual age of the tissue or where the prey are located. But they're laying their eggs on the bottom of the plant. And the way in which these crops are grown, they are very heavily pruned and maintained on vertical trellises. So farmers are going in there and they're pulling off these bottom leaves 1 to 2 times a week, depending on the crop growth rate. So we really learned that they are potentially removing that agent that they're paying to put into the tunnel. The other thing that we saw was some negative impacts as a result of screening. So in addition to reduced retention of some of our natural enemies, we saw a lot of flower and fruit abortion. And that was because of the increased temperatures as a result of blocking that airflow or that ventilation capability in that tunnel. And so like I mentioned, we used a very, very small mesh. And what you're seeing here are the mean daily maximum hottest temperature that was recorded in these tunnels. And the solid blue line here are open tunnels with ventilation sort of at its maximum. And then the red here are the screened tunnels where we were really limited by that material. And the green shading here is the optimal temperature for tomato production. So you can see the majority of that growing season, temperature exceeds the optimal temperature for crop production. We then went back into the lab and conducted some controlled experiments in growth chambers to see how that temperature impacts the survival of lacewing eggs. And so here we worked within controlled environments where we just purchased lacewing eggs, and then we placed them in these held temperatures and looked at the viability of the eggs that were hatching. So on the right-hand side here, it's the number of viable eggs per female. And on the left, when we reared adults in there, it's the number of eggs total that they laid. So you can see the optimal temperature here for oviposition and egg hatching for lacewings is around 17 C. And as we get into those hotter temperatures that are experienced in the growing season in high tunnels, we exceed the temperature at which this insect can grow and survive. And so from this work, recommendations were developed that suggest that lacewings and minute pirate bugs are potential predators that could be incorporated into high tunnels. And oftentimes we actually do see them naturally moving into these environments. Those herbivore-induced plant volatile lures and floral resources can enhance this retention. So it can be of value to diversify your crop, in particular in terms of pest management. And intense pruning and lacewing biocontrol may not be compatible. So intense pruning is often necessary to mitigate pest disease, especially disease as it's related to high humidity. So some of the powdery mildew. So if you are not able to leave the plant tissue in there longer for those eggs to hatch, then you would have to rely on another natural enemy to contribute. And we did see some benefits of screening, but we need to increase the ventilation. And so when I talk about benefits of screening, this moves into the second grant that I got to work in high tunnels. And what we noticed was really increased production of our cucurbits in our screened tunnels. And so this next part here was a grant that I got from NIFA as a postdoc, actually, here at Purdue, where we went in deeper and looked more at the effects of screening. So when it comes to cucurbit production, one of the biggest challenges for growers to manage here in the Midwest is a pathogen that we refer to as bacterial wilt. It's called Erwinia trachyphylla. It is a pathogen that is transmitted by the striped cucumber beetle, Acalima vittatum, primarily, and some other beetles that infest these crops. But so the beetles move into the high tunnel, they come out early in the spring and find whatever cucurbit they can, and they begin to chew and feed. So if that's a very small seedling, a new transplant, they can defoliate the whole plant. If it's a larger plant, they feed in these holes, and then the bacteria resides in their salivary glands and in their frass. And as they feed on these, they transmit the bacteria, which resides in the phloem, and it blocks water transport and leads to plant death. And there's no sort of recovery from that. And what we noticed was that our tunnels that were screened had no bacterial wilts. So that was something we wanted to investigate. So we went back to this research farm. We focused on growing some specialty cucumbers and some specialty melons, very small melons in these high tunnels. And we imposed on these tunnels 2 larger sized screening. So here the hole is 1 by 4 millimeters and 0.72 by 0.97 millimeters. So much larger, sorry, the last one was 0.04 by 0.045 millimeters. So much larger mesh size. And these sizes were taken, chosen based on what was commercially available and the size of the beetle vector we are trying to keep out. And so we put this screening on 2 high tunnels. We have 6 total, so 2 with the large screen, 2 with the medium screen, and 2 that were open. And then we looked at our cucurbits. And so what you're seeing here is the number of striped cucumber beetles per plant. And we did this across 2 years. So this is the small screen that was done in the first study. And like I mentioned, complete exclusion of cucumber beetles. When we increased that screen size, we did see that some beetles are getting into the environment. So open tunnels have no screening. The intermediate screening here has the lowest number of beetles, and then the large screening definitely let the beetles in. The economic threshold for striped cucumber beetles on cucurbit and cantaloupe production is 1 beetle per plant. So if we can keep that threshold, the number of beetles below that threshold, that's really promising. And so we were excited to see that that intermediate and actually even open fields kept the beetles below that threshold, but especially in the last year, that larger screen actually kept them inside, and then they reproduced and increased in numbers. And so one of the secondary things that we noticed in this situation with screening was that we had severe increase in the number of aphids per leaf. So looking in 2016 here, open tunnels had about 2 aphids per leaf on our cucurbits. Where we got to that medium-sized screen, it was up to 12 aphids per leaf, and the large screen was somewhere in between there. And this kind of went along with what we saw the first year when we used the really small screening. So in the year that we had the flower and the volatile lures, we saw a dramatic decrease in the amount of natural enemies that are colonizing these tunnels on their own, right? So while trying to keep the natural enemies that we released in, we also excluded all of those that would naturally find their way. And most of these consist of spiders, syrphid flies, a variety of parasitic wasps, and buridids or stilt bugs. And what we did notice was that we saw an increase in the colonization when we diversified that crop. And so here we were able to optimize the screen size to manage cucumber beetles and bacterial wilt, but secondary pest outbreaks really remain a challenge with that interference of natural enemies being able to move into the system on their own. The next part of this grant was looking at growing unique cucurbits. So this is something that has become increasingly adopted in the United States, but globally is much more popular, and that's the idea of grafting annual plants. So I thought that this was a good system to look at grafting impacts on insect pests, and that's because the susceptibility varies among crop types in relation to cucumber beetle damage, and in particular, the bacterial wilt pathogen that they transmit. So watermelon is resistant to this pathogen. We find some resistance among winter squash and pumpkin, but cantaloupe and cucumber have no resistance. And we have seen that resistance can be achieved through grafting to some insect pests. In particular, Colorado potato beetle, when grafting different solanum together that vary in their resistance. Grafting citrus to control two-spotted spider mites, and grafting chrysanthemums to control thrips. So what we wanted to do here was take the scion, this would be the tasty variety of your choice, in particular, which cucumber or cantaloupe variety that you would want to grow in your high tunnel, and grafting that onto a rootstock that is resistant to the herbivore or the pathogen. In this case, the bacterial wilt that the beetles are transferring. So we were trying to graft cucumbers and cantaloupe on top of watermelon or squashes and gourds, which are not susceptible to the pathogen. And so we began by evaluating these in the greenhouse. And so we removed the insect in this situation, and we only worked with the pathogen that that insect is transmitting. And so we grew some cucurbits in pots. We used this thing called a floral frog, which has a whole bunch of pins on it. And we would push that into the leaf to create wounds. And then we had the bacteria that we were growing in culture and broth media, and we would pipette that on and sort of massage it into the leaf. And then we let those plants grow, and we evaluated the time until the first symptom of wilt appeared and the time until the entire plant succumbed to the wilt. And so when I display this information here, I want to orient you to the graph. So across the x-axis here, we looked at 3 different resistant rootstocks. So they're called R1, R2, and R3. And this line here is showing you the bottom portion of our plants. On the y-axis here, we have the number of days until that plant died. So what you can see here is that our resistant rootstocks did not succumb to the bacterial pathogen and grew for the entire duration of the experiment. Then what we have over here is the susceptible scion. So this is a cucumber here, S1, and S1 grafted onto itself as a control. And what you see here is that that plant died within about 15 days of inoculation. Then if we look at these 2 different graft combinations. Here we have this cucumber scion onto rootstock 1, rootstock 2, and rootstock 3. We see that all 3 of those plants do eventually die, but it takes longer on rootstock 2 for those plants to succumb to complete wilt. We then repeated this with a cantaloupe variety onto rootstock 1 and rootstock 2. And what you see here again on rootstock 2, we see this prolonged time until complete death, but all of the plants do succumb to wilt. Then we took these grafted plants and we put them out in a high tunnel and in the fields to evaluate their field level susceptibility to both the vector and the bacterial pathogen itself. And so same orientation for the grafts here. We have 2 different scions that we're investigating, susceptible 1 and susceptible 1 onto rootstock 1, then susceptible 2 and susceptible 2 onto rootstock 1. And here we're looking at the number of cucumber beetles per plant. And what we see here for the first susceptible variety, we see a reduction in the number of beetles per plant when it's grafted onto rootstock 1. When we go over here to susceptible variety 2, we see no difference in the number of plants, beetles per plant. So really mixed results, not too much in terms of the possibility of implementing grafting as a way to manage cucumber beetles or bacterial wilt. But we also saw some other really interesting things that we think may be worth pursuing in the future when we did these field trials. So again, here you're looking at those 2 susceptible varieties on top of the 1 resistant rootstock. And here we're looking at 2 different pests. So the top graph is spider mites. And here we're just counting the accumulated number of days in which the plants were infested with spider mites. And the same thing down here, looking at powdery mildew. So the accumulated number of days in which those plants were exhibiting symptoms of powdery mildew infection. And what we found for the susceptible variety number 1 grafted onto rootstock 1 was actually an increase in the susceptibility to spider mites. So when that plant was grafted, we saw more spider mite damage than compared to the non-grafted. And we saw something similar with the response of that cultivar and graft combination to powdery mildew, an increase in susceptibility as a result of the graft. If we look at 2 other pests that we encounter in this system, here on the right, we have top right is accumulated number of aphid days, so days in which those plants were infested with aphids, and the accumulated number of days in which we saw thrips occupying those plants. And in this situation with the susceptible variety number 2, we see increased resistance, fewer aphid days when it's grafted onto rootstock number 1, and fewer thrips days when it's grafted onto rootstock number 1. So what we really conclude here is that grafting can reduce the onset of symptoms and time until death, but this really isn't a means to combat the pathogen, bacterial wilt, or beetle damage in the field. It's not at a level that's suitable for growers. Grafting did reduce the number of beetles per plant, but again, not to a value that would be acceptable. We do see that this interaction is very scion and rootstock dependent, and there are potential impacts on other plant pest interactions that would be interesting to pursue. In some cases, grafting can actually make that plant more susceptible, which is what we saw. So to sort of round out where we're at this point with high tunnel production, we've been able to increase our knowledge of insect dynamics in high tunnel systems, really demonstrating that they're a unique growing environment that experience problems that are different than field production and greenhouses and can sometimes be exacerbated. We evaluated some strategies to manage insect pests and were able to identify a few potential natural enemies that may be compatible with high tunnel production and suitable across seasons. Currently, I just received funding to continue to work in this system. And here we're going to continue our work with cucurbits. So I've been a part of a grant that's looking at grafting for managing cold tolerance in the roots to grow cucumbers earlier in the season. And we've had some interesting observations in terms of mites through that work. And so we're going to be expanding that. So two-spotted spider mite is the second most devastating behind cucumber beetles. And so we've been able at our research farm to screen our tunnels and manage cucumber beetles. And now we're trying to get after spider mites and improving our management capabilities of this pest. And so what we have seen so far in comparing different cultural growing methods is a difference in the ability of this pest to disperse and how quickly it moves across the crop. So we were looking at using this netting system in which the cucurbits are grown at the about 12-inch spacing, and they crawl up this net and sort of create a wall. And in this system, we don't prune, and we just sort of let the plants go. And once they fill the wall, we'll cut the top of the vine, but the runners continue to grow and support themselves. It saves on labor, but it reduces airflow, and we see mite infestations exacerbated as they spread across this system. The alternative is the 1-liter system, which is very, very labor intensive. So we space the plants out a little further. So they're at minimum 18 inches in this system. And we prune all of the side shoots and runners, and we maintain one stem, and that stem is allowed to continue to grow and trellised on a vertical wire. And as the plant grows and reaches the top of the tunnel, we harvest the bottom fruits and we prune it and we wrap that stem so these plants can be put into the high tunnels here, usually late March or early April, and remain until we get our first really hard frost into October. So they're very long-lived. And in this situation, we see that the mites don't move as quickly and spread as quickly inside of the high tunnel. What I'm really interested and excited to start working on this year is developing a monitoring protocol to aid growers in early detection. So at this point, oftentimes we see that growers don't intervene and try to do anything to control mites until they're at the point where they're finding the webbing and silking on the plant, which is often too late. So I have a new PhD student who's going to be working on developing an early monitoring and early detection protocol that's easy for growers to implement. And then looking at, this is the most exciting to me, that natural enemy compatibility and high tunnel production. So we have a wide variety of predatory mite species that are commercially available. Some of them are generalists and some of them are specialists, in particular for two-spotted spider mites, which is our pest here. But they all have a different ideal temperature for where they're actively foraging and feeding, and a different relative humidity under which they perform best. And so in this environment where we don't have a lot of control over temperature and humidity, and we know that it varies drastically, even from morning to nighttime or from early in the growing season to midsummer to later, I'm really excited to look at which of these species can disperse and move vertically throughout the crop, as well as tolerate the environmental conditions under which, so that we can develop recommendations for the best predator or combinations of predators to release in high tunnel systems. We're also going to be looking at evaluating biopesticides. So in particular, some of the newer products that growers are interested in utilizing include Chromobacterium subsugi, heat-killed Burkholderia species, and Bavaria bassiana. So we're really interested in looking at what is the best method for delivery of these products. So what we've been using most recently is an electrostatic sprayer to apply these in the high tunnels to get really good coverage. But we also want to see how these products are compatible with the natural enemies and the extreme environmental conditions that growers face in high tunnels. And the last bit that I want to talk about is moving into hydroponic production systems and really, really controlled environment, oftentimes relying completely on supplemental lighting to grow crops. And so indoor vegetable production has really been changing and been really innovative in recent years. Growers have had more access to LED lights to save on energy costs. And with that technology is the ability to customize the light quality or the color of that light and manipulate the growth and quality of the plant. Hydroponic systems are also being integrated because they are much more water efficient. And in this situation where it's often a warehouse or an old industrial building that's being converted, we're able to grow year round and accelerate crop growth. So get to a mature stage much more quickly. This market right now is dominated by a lot of leafy green and lettuce production. So what I'm really interested in understanding is how light quality and quantity influences plant chemistry. We have seen some situations where you can get protection from plant pathogens by changing the color or the quality of the light. But how does it impact plant chemistry and that interaction with herbivores? So I was really fortunate to work with Dr. Krishna Namali, who is here at Purdue in the horticultural department, and Dr. Rehan Mohammed, who came here as a Borlaug fellow. And so this was my first venture into this, and we started working with hydroponic lettuce. Rehan really wanted to learn how to implement hydroponic growing to help water saving. And I was really interested in the lighting technology and the pest management. So we put all of that together and had her work really hard for 12 weeks to learn some new things. And so we looked at 4 different varieties of lettuce, and we compared their production under LED lighting conditions with high pressure sodium, more full spectrum lighting conditions. We didn't see a big difference in the overall quality or production, so very similar biomass production in these 2 systems. We also worked with the green peach aphid in this system, and we caged these aphids on these plants, and then we put some natural enemies into the system as well to see how these different lighting treatments would impact. And I think some of the most exciting results from this, it really boils down to plant tolerance or resistance in this case. So here what we're looking at across the x-axis are the different treatments that we implemented. So here we have a negative control. So we are infesting the lettuce with aphids and we are not doing anything to control that population. Then we have LED lighting in place of the high pressure sodium. So a more narrow wavelength and color of light. Then we released lacewings and ladybugs under the traditional lighting system. So high pressure sodium, just looking at how they can move across the tray and spray the aphids. And then we combined the ladybugs with this LED lighting to see how does that light impact the ability of this predator to forage in this system. And our sort of positive control check here was applications of permethrin. And so we have different colors to indicate the different types of lettuce. We had a butterhead lettuce, an oak leaf lettuce, which is a red leaf lettuce, a romaine lettuce that produces red leaves, and then green leaf lettuce. And so what I'm showing you here is the change in aphid population over time. So we infested these plants with a certain number of aphids. And if the bar goes below the line, that means that that population was reduced. It went down over time. If the bar goes above this 0 line, that means that that aphid population increased over time. And so one of the first things we were able to identify is this resistance of the butterhead and the oak leaf variety to aphids. So we saw that regardless of any control where we did nothing, those populations declined over time. So that variety is not suitable. When we look at the susceptible varieties, so in this case, the romaine and the green leaf lettuce, we see something different. And so first I want to point you to just the light alone. So just changing the light quality in this instance decreased the aphid population growth over time. So while the aphids are still increasing, it's at a much lower level than it is compared to those under the full spectrum light. Also, here is an opportunity to look at how LED light impacts the foraging ability of ladybugs. And what we see here is no difference. So in this situation, the LED light did not inhibit the ability of these larvae to forage and find prey. And what we saw lastly, what was interesting to me was that the LED light treatment did not differ from our chemical application. So we were getting as good of population control by applying permethrin in this case as just producing the plants and the insects under the different light quality. We then went into the lab and isolated just the lights and the herbivores to look at the impacts on predator behavior. So we did this in a bioassay, like I mentioned, in the lab, where we put a leaf disc random movement, which the predator is just moving all over in the disc, movement towards the prey item, or actual contact and consumption of the prey. And so we measured every minute for 20 minutes. We looked at 5 different light sources and conducted 16 replicates. So here's the lacewing larvae, the lady beetle larvae, and this is what our setup looks like. And so what you see here are the different light treatments. So we expanded the colors that we were looking at. So full spectrum control, purple, blue, purple, and red incandescent. And what you see here is that we noticed with the lacewing larvae, we saw less time of the larvae in contact and consuming their prey when they were foraging under purple LED and red incandescent. And then after Raham left, I was hired as faculty and had an opportunity to invest in some behavior monitoring technology. And so now we have moved on to doing these assays under a video camera, and we use this EthaVision software to analyze the behavior of the predators. And so we can designate in this software where the zone of the prey item is and where the predator is, and it can track its movement, its velocity, and its time in particular areas. And what we saw here, looking at lady beetle larvae and lacewing larvae, was that if we look at first on the left, the mean mobility, so just how much are they actually moving during the observation period, we see a suppression in the mobility of these insects under red light, which is what we would expect, because red light typically insects cannot see in that light, and so it arrests their movement. But what was interesting along with that was that we saw them spending more time in the zone where the prey items were. Compared to other light treatments. And then if we look at the purple light, which was the other light we saw responses to when we were doing this visually before the software, we see a little bit of an increase in their mobility, but not necessarily different from the other light treatments for the lady beetles, but more so with lacewings. And so this may suggest that they're just moving around and disoriented because we don't actually see them consuming prey in that situation. And so this just gives you an idea of what the difference in the quality of light or the wavelength looks like. So this yellow line is our control high pressure sodium. The red incandescent light here has nothing down on the 400 zone of the spectrum. And then the purple one is very close to blue, but has slightly less red. So very, very minimal changes in the light quality. And lastly, this is sort of the direction in which my light work is moving. So hemp or cannabis sativa is an emerging specialty crop. It's become very popular and was recently nationally legalized. And so this is another crop that's moving into these controlled environment and artificial lighting situations for production. So I'm really interested in understanding the interactions between light, plant growth, and insects to identify pest management strategies. And so I received some funding from Purdue in an ag seed grant to work on this. And so some of the largest pest challenges for this crop, particularly in controlled environments, is the hemp aphid or the cannabis aphid, the hemp russet mite, and two spotted spider mites. So the hemp aphid is extremely prolific and can cover the plant and completely decimate it, although we don't know what the economic damage looks like. They can survive on those plants for a very long time under obscenely high numbers. And the same thing with the mites. They preferentially colonize the bud or the flower of the plant, which is the part that you're harvesting, but we often find them on leaf tissue as well. And so the challenges that come with hemp production is that it is a highly regulated crop. As of now, we only have a few biopesticides that are allowed as a pesticide that can be applied to this crop to control pests, insects in particular. And there's really strict regulations around the chemical composition of the crop, particularly the amount of THC has to be below a specific level, and it's really unpredictable in terms of what influences that. And if the THC level goes too high, growers have to destroy the whole crop. So what I'm really interested in looking at is light quality and how that impacts the THC composition and aphids and mites, and then again cascading up to natural enemies, generalists and parasitoids. So I have a new master's student who's going to be continuing looking at different light wavelengths and red to blue ratios, how that impacts plant chemistry and these pests and natural enemies. But in the meantime, we've looked at biological control and using a very common greenhouse banker plant system and implementing that into indoor hemp production. And we evaluated 3 different parasitoids, Aphidius colemanni, Aphidius ervi, and Aphidius matricarii. And from this initial work, we would recommend Aphidius matricarii and Aphidius ervi in these banker plant systems. And so with that, sorry, I'm going to skip to the end and take any questions. Oop. Thank you very much.
Thank you دكتور لورا. أي أسئلة يا جماعة؟ هو سؤال واحد بس عشان الوقت. في أي أسئلة؟ Okay. Thank you, Dr. Laura, from United States of America, University.
دكتور أمال هتسأل سؤال.
Thank you.
You're welcome.
دكتور أمال هتسأل سؤال.
طب sorry, 1 minute. Yes.
سؤال.
اتفضلي دكتور أمال.
دكتور أمال.
أو دكتور أحمد.
Thank you, Dr. Thank you, Dr. Laura. It's not asking, but I didn't catch what is the relation between the variety of the plants and the numbers of the beasts, as you said. Actually, it wasn't as I imagined.
I'm sorry, what was that? The variety of the plant and
the numbers of the beasts.
the numbers of the beetles?
Yes.
Yeah, we saw, regardless of any of the grafting combinations, we never saw an impact on the number of beetles feeding on those plants. So we also had some more I didn't even share with you today, but we never saw a difference of grafting impacting the number of beetles feeding on them.
Yes. Okay. Thanks a lot. Thanks a lot.
Okay.
And I see a question in the chat there about how does hemp affect insects at a molecular level? I don't know that anyone has looked at that on how the secondary compounds in hemp impact insects. We have 2 insects identified that are specialists on that, and maybe they can take advantage of it, but we also have a very large suite of generalist insects that have no problem feeding on them. And I put my email in the chat if anyone would like to reach out.
Yes, okay. That's thanks.
Okay. Thank you so much, Dr. Laura. دلوقتي معانا إن شاء الله آخر محاضرة مع دكتورز أجرين أند مارجريت أند بروس من الولايات المتحدة الأمريكية جامعة فيرمونت. هيتكلموا معانا عن دور الأشعة فوق البنفسجية في المكافحة المتكاملة للتريبس في الصوب الزراعية. دكتور أجرين, are you here? Dr. Agre?
Hello?
Yes.
Can you hear me? I'm sorry. Something is wrong with my headset. Okay. Can you hear me now?
Yes, yes.
Okay, perfect. So I'm going to share my screen. Okay. Here you are. So hello, everyone. Thanks for having me here today. I'm so excited to share with you some of the very interesting results of our current project. In this project, we look at the effects of UV light for integrated pest management of Western flower thrips. This work was initiated 3 years ago with a funding from American Floral Endowment. And we started a collaboration with a lighting center in New York, who are a leading center for lighting research and development. So we, Entomology Research Lab at the University of Vermont, provided the entomological part of this research, and they helped us with the UV light part of it. So let's begin with an introduction of Western flower thrips. Thrips is a small insect which causes a huge damage to several species of vegetables and ornamental crops. It's a very tiny insect. Adults are in the size of 1 to 1.5 millimeter, and larvae are also smaller, even less than 1 millimeter. These insects cause huge damage by piercing its mouth part to the cells of the plant and suck up the content of the leaves. It also, by this feeding behavior, it causes a huge cosmetic damage on the leaves that are basically like silver patches and some, black dots that shows the, excreta that indicated the presence of the insects on the plant. Beside the direct damage, this insect also transmits some important plant viruses, such as tomato spotted wilt virus and also impatient necrotic spot virus that are both very important in terms of causing, diseases to especially tomatoes and cucumbers. So this insect is very difficult to manage basically because it's very small and it can hide through the crevices and underside of the leaves. So it's really hard to target this insect with pesticide. It also has a very rapid population increase. So the whole generation can be completed even less than 30 days, which is a very fast reproduction rate. There is a wide host range for this, pests from all many different species of ornamental and vegetables. This insect is also parthenogenesis, meaning that males can be, hatched from unfertilized eggs and are haploid, and females are diploid. So, even like females can even cause like damage and, lay eggs without the presence of males. These insects also develops a very rapid resistance to chemical pesticide. Right now, there is more than 30 active ingredients that this insect has been developed resistance against. So the UV light is extensively used for sanitation purposes, and it has been reported that it has a high germicidal effect. This effect is particularly in the range of 264, the wavelength of 264, which is basically UVC. And this is a very high energetic range. And these are like short wavelengths, and they are very highly energetic. And this part of the UV range is usually absorbed by the oxygen and ozone in the atmosphere, and we don't get much of it in the terrestrial sunlight. Which is a good thing because it's very energetic and it causes huge damage. It can also penetrate to the outer membrane of the organisms and cause chemical damage. In some cases, it has been reported that it causes a mutation in insects, and it can change their behavior in terms of, feeding and navigation, and also finding sex. So the smallest the insect is, it's more susceptible to UVC. And we actually took advantage of this fact, and we started to see if UV can actually have any effect on thrips. There are some studies that shown the effect of UV light on some plant pathogens. And there is a research team in Cornell University that they looked at the effect of UV on powdery mildew and also downy mildew in strawberries and cucumber. And they could see that by increase in the dosages of UVC, there were less spores that could actually germinate from the surface. So that was a successful result showing that, okay, so higher doses of UV can actually eliminate the spores from germination and growth. So having that background information, we started to develop a research considering how UV light can, first of all, directly kill the insect. So we started with the several questions. First of all, we wanted to know if there is any lethal effect of UVC against Western flower thrips, adults, and larvae. We also wanted to know if UV can change the number of eggs that are going to lay by the females. And also, can we actually target the eggs inside the leaves? Because thrips usually lay eggs inside the plant tissue. And it's really hard to target eggs because of this fact. So we wanted to know if we can actually target those eggs inside the plant tissue by different dosages of UVC. And we also wanted to know that, the important part of this research was that even if we came up with some good dosages, we needed to make sure that those dosages are not going to damage the host plant. Because we had this in mind from the beginning that this project is going to end up in some, machinery or system that could be applied inside the greenhouse. So we had this idea that UV light could be emitted to thrips while they are feeding on the plant canopy. So we don't want any damage from the UV on host plants. So we wanted to test that one too. And we also wanted to come up with a machinery or, applicator kind of thing that we could apply UV inside the greenhouses. So here is a chamber that we use to run most of our experiments. As you can see, that's a huge chamber, and there are UV lights installed in the top of the chamber, like right here. And there is a shelf right inside the chamber that we could adjust the distance of our samples from the light. There is a reading system outside. There is a sensor and a switch that we could actually turn off and on the lamp anytime we wanted. And for safety purposes, there was also a shutter that could be closed while the door was open. So this screen that you see in this picture, the black box, actually gives us the amount of irradiance that was coming out from the UV lamp. And we needed to kind of calibrate and come up with some dosages that we wanted to use for this experiment. So UV is basically the product of intensity and time. The intensity was kind of constant, and it was whatever coming out from the lamp. But we could actually play with the time factor of it. So we, played with the time factor a little bit, from like 1 second to 30 minutes. I mean, we tested a huge range of exposure in time to come up with the best dosages. This actually took a couple of months so that we could feel comfortable coming up with some good dosages that could possibly impact thrips adults or larvae. So that part is basically like calibration and coming up with the best UV dosages that could possibly have lethal effect on the insects. Here is our thrips rearing diagram that it shows that we actually keep our thrips, we rear them on bean leaves, and the colony is kept at 22 degrees Celsius and with humidity of 75% and 16 to 8 lighting and dark condition. We keep those bean leaves in glass vials with water, and we place those tubes in some plastic containers, keeping them in wooden chambers. And the reason for keeping this going colony was we always want to test the same age insect, whatever we wanted to test against them. We always wanted to have even age individuals, so we could actually control part of the error. These are bioassay chambers. We had insects in petri dishes, and after every exposure to UV, we would place a piece of leaf for them, and we would just incubate them at 25 degrees Celsius and the same lighting regime, and we would do our counting afterward. So here is the first graph that shows the LD50 of larvae and thrips larvae. So as you can see, we tested different dosages. To begin with, we tested 5 different UVC dosages, and of course, we have also controls. So after exposure, we kept the thrips onto 5 days. So we did the counting at day 1, 24 hours, actually it's 1 hour after exposure, 1 hour, 24, 48, 72, and also 120. So we kept counting the mortality after exposure to UV. And as you can see, the mortality started to increase over time. Only after 1 hour after exposure, you see the highest dose, the red line, showed higher mortality compared to control and also lower dosages, and this could increase over time. So after 24 hours, we could actually target 50% of the population with the highest dose. So we also did the same thing against adults, but we had to pick a higher UVC dosages. And the reason is that adults have a kind of impermeable exoskeleton, and they are more mobile. So the thicker exoskeleton and also being more active might be some of the reasons that they were not as susceptible as the larvae. So we increased the UV dosages that we wanted to apply against larvae. And as you can see, we picked the higher dosages, 5 different dosages and controls. We got the same trend, but as you can compare, if I'm going to go back to the previous slide, as you can see, the mortality range was lower in terms of, in case of adults. So we had 50% mortality after 72 hours at the highest dose, which was even hardly 50%, was even less than 50%. So that was, so doing that, we could conclude that, okay, with direct UVC, we might be able to actually target almost 40% of the population. And we cannot, with these dosages, we cannot actually target the whole population. And we didn't want to go like further. We didn't want to test higher dosages, because higher dosages means that longer exposure time. And in terms of like practical point of view, when growers are going to use this UV technique inside the greenhouses, long time is very important. They're not going to stay there and apply UV on the trips for 1 hour or for 45 minutes. So we needed to be careful, not exceed with the dose and also exposure in time. So we decided, okay, so we got like, we could target 40% of the population. How about the 60 population that we couldn't kill right away? What happened to those population? So we exposed Western flower thrips adults to different UVC dosages. These are actually sublethal dosages. And we wanted to see if UV can actually make any effect on the number of eggs that are laid by those insects. So we actually selected three dosages, and we exposed that 60% survivors to this dose. And we exposed them to UV, and we allowed them to lay eggs for like 24 hours. And after that, we counted the mortality up to 12 days. As you can see, after day 7, we started to see a difference between the number of eggs that hatched from exposed adults at different dosages. So this star sign shows the control, and the higher dose, which was 3.5 joule per square centimeter, started to have the lowest number of hatched eggs. So we counted actually the number of hatched eggs over time, and we realized that there was a significant difference between the number of eggs hatched between controls and treated adults. So it was actually interesting, because we could realize that the average number of eggs that hatched from control adults were like 30 eggs, but also from the exposed adults were almost 12 eggs in average, as you can see. So that was a good result, and we could realize, okay, so there might be some sublethal effect of UV on Western flower thrips, not only like killing the insect right away, but we might be able to actually change the fecundity of the insect by UVC. The other sets of experiments that we focused on later was exposing the leaves that had eggs in them to different sublethal dosages of UVC. So we exposed the leaves to Western flower thrips adults for 24 hours, and we had them lay eggs. After 24 hours, we removed the adults, and we exposed those leaves that had eggs on them to different dosages of UVC, because we weren't sure that if an insect is going to lay eggs on the surface or on their bottom side of the leaves. We did this experiment both on the leaf surface and also underside of the leaves in two separate sets of experiments. And as you can see in this graph, by increase in UVC, we had less hatched eggs from exposed leaves. And it was really significantly different and lower than the number of hatched eggs from the control. Doesn't matter if it was like the surface of the leaf was exposed or underside of the leaf. In both cases, we had significantly lower larvae that hatched from the exposed leaves. And the reason that we counted the first stars or the larvae of the thrips was that we weren't able to actually see, because thrips eggs are so tiny, so they are not really easy to be seen, even using a microscope. So we had to do everything based on the number of larvae that are hatched from the leaves. We did these trials on bean plants, as we always, run our first, trials on bean plants, and we also test the marigold plants with the same procedures, exposing leaves that contained eggs to different UVC dosages, and we got the same result, even less eggs hatched from exposed leaves compared to control. So we not only could like control 40% of the population of the adults, those 60% survivors could actually be affected by UVC by not laying as many eggs as the controls, and also even leaves that had the eggs inside them were impacted by UVC and less eggs could hatch. The next objective, as I mentioned earlier, was make sure that those UVC dosages that we selected are not going to damage the host plant. So we picked 5 different plant species that are mainly used in greenhouses. These 5 ornamental crops were exposed to 3 main UVC dosages that we have been testing. And the good news was that no damage was actually observed after exposure to UVC. There was only like minor discoloration in some species like mums and roses, as you can see in this picture, just like tiny, discoloration at the edges of the blossoms. But this was fully recovered after 3 weeks, and it could make the plants totally saleable and looking normal. So these actually changes faded after 3 weeks. The other species that was a little bit susceptible was portulaca. As you can see, 3 days after exposure, there some of the leaves fell, and some disorientation and some changes happened to the shape of the plant. So this is control, and as you go from this profile, you can see like by increasing the dose, there are some damage. But as you can see over time, after 1 week, after 2 and 3 and 4 weeks, you can see that damage was partly removed and made the plants look almost normal. This was one of the plants that at the immature stage was susceptible. And as I mentioned, mums and mini roses were a little bit susceptible at flowering stage. But again, those issues resolved after 3 weeks, which was good. So we could feel assured that, okay, so the dosages that we are going to test against thrips are not going to damage our host plant. So I think the overall conclusion of this project so far is, imagine that you have 200 Western flower thrips. You treat 100 of them with the highest UVC dose, which was 3.5, and you don't treat the other 100. Those control hundreds, each of them will lay 30 eggs in average, which results in 3,000 of individuals. On the other hand, exposing those larvae to UV will impact 40% of them. Out of the 60% of the survivals, each of them, based on the egg trials, each of them will lay 12 eggs on average through their lifespan. And this comes up with 720 compared to 3,000 under control condition. In addition, we treated some adults by UVC, and we realized that they started to lay 76% less eggs compared to control. And 96% fewer eggs was also hatched from the leaves that were exposed to UV. Doing some calculation here, it shows that the results of this exposure to 100 individual will result in 28.8 eggs would hatch from the whole population of that 100 adults, whereas in control we'll have like 3,000 individual. So considering these two ratios, we could actually control 99% of the whole population, combining all these 3 methods, like targeting adults directly by UVC, impacting their fecundity, and also targeting eggs inside the leaves. So considering all these 3 experiments that we did individually, if we just want to come up with a result, we could say, okay, we were able to control these 100 individual like 99%, which was great. I mean, considering okay, I understand that this might be under, highly controlled condition, but this is not going to be our end of the project. We are definitely going to move on and test everything in a real world. So right now we are focusing on the whole plant canopy. So far, most of our experiments was in Petri dish scale with trips and leaves inside the Petri dish. But right now we are looking at the whole canopy of the plant and see, okay, if we have a UV lamp as a source on top of the canopy, how much of the UV dose we can get at each level through the whole canopy, and how much of that control we can have through this whole system. And once we got those data, the next and last step is to actually coming up with a form of applicator. We are right now working with a mechanical engineering department, and we are working on developing a device that could actually transfer this UV technology into the greenhouses. We would definitely consider looking at the effect of UV light from the top of the canopy and also from the side. So we will have like UV applicator going through the rows and emit UVC through the whole canopy. And we will do, we'll see how this could actually affect the population compared to control condition. So we are so excited about this part. We are working on that right now. So far, we have been working on like some basic of this research. I have to say that this is one of the very first studies looking at the effect of UVC on pests in greenhouses. Most of the work has been so far on like pathogens, on plant pathogens, but not really much has been done on insects. So we are so proud and we're so happy that we're getting perfect results and we're gonna move on strong. The other project that is kind of like parallel with this UV project is that there are studies that show that UVC can actually induce some plant defense mechanisms inside the plant. So once we expose plants to UV, there are some defensive enzymes and secondary metabolites that could be induced inside the plants. Most of them are actually controlled by phytohormones such as jasmonic acid or salicylic acid or ethylene. These are all induced the plant defense against herbivory. So one of the other projects that we are working right now on is that we are going to expose some of the different species of ornamental plants to different UVC dosages. And that is probably like more than once exposure. We're gonna just expose them to lower dosages, but over time, probably like over a week, we're gonna expose them once a day. And then at the end of the week, we're gonna do some analysis on the chemical compound inside the leaves. We're gonna use an NMR technique at the chemistry department. They have the facility, so we can actually look into the kind of like profile change of this chemical compound inside the leaves. And if we could, actually make some of those secondary metabolites activated inside the plant, there is the hope that that could actually make those plant resistant against herbivory, especially against thrips. So that's the next thing that we are going to test, and we're so excited about that. By saying that, I'm reaching to the end of my presentation. I hope you enjoyed it, and I would be happy to answer any questions that you might have.
Thanks, Dr. Argen, for that great presentation. I want to ask you a question. Can this method or ultraviolet be applied successfully on many insects than Frankinella occidentalis of the greenhouse?
So was your question that we can actually apply this system against other insect pests?
Other insects than Frankinella occidentalis?
Well, we would love to. I mean, we just started this project with focus on thrips, But we are definitely considering, if we come up with the whole protocol, we're going to consider looking at the effects of this UV lamp on other insects and also other components of an IPM system that we are going to have. So we're going to make sure that these UVC are not going to have any harmful effect on natural enemies or some endophytic fungi or endopathogenic fungi that we use in the system. So the next step is actually, one of the consideration is actually make sure this UV actually can be a good fit inside the system.
Any questions?
We can Dr. Argen, there is a negative impact on field crops or yield by using the ultraviolet?
Oh, that's a good question. So far, we only looked at the effect of UV on like appearance of the plants, because our focus was ornamental crops, and we were only interested in looking at like the appearance and not having any cosmetic damage on the plant. But that's a very important thing. If we want to use UV system against, for example, vegetable crops, that is something very important to consider, that we need to look at the yield of the crop. We have not tested UV against vegetables or other type of crops. We have been focused only on ornamentals. But that's a very important thing to consider.
السلام عليكم. السلام عليكم.
اتفضل دكتور محمد. اتفضل دكتور محمد.
Please, I wanted to ask about the cost of use of UV to control syrups in one greenhouse area about 1 hectare.
Well, as I mentioned, it's one of the first studies that we are looking at the effect of UV. It has been basically like lab trials so far and running trials under controlled condition. In the next 3 years, we are going to definitely focus on cost calculation and the way that we can actually transfer this technology to greenhouses. So we are really at the early, stages of this process and this project. But I'm not sure how much it's going to cost. It's definitely not going to be very expensive because the UV lamps itself are not that costly. The important thing is the best way and the best system to actually apply this technology. We have some concerns, and growers obviously will have some concerns about the safety aspect of application because nobody wants to be exposed to UV. So we are thinking of some methods that we can actually protect growers from applying this technology. We might have some hot spots in greenhouses, and we could only activate UV light during night that nobody is technically in the greenhouse. So these are all the considerations that we are taking in mind and we will address in future. So hopefully next time when, if I had a chance to, share our results with you, we'll have more information on the economic aspect of it for sure.
Anyway, thank you.
Thank you, Dr. Irene.
You're welcome.
Dr. Marguerite Adley, you can ask.
دكتور مارجريت عدلي، ممكن حضرتك تتفضلي بطرح سؤالك.
دكتور أحمد المرسي.
دكتور أحمد المرسي.
السلام عليكم.
وعليكم السلام، اتفضل.
Thank you so much for the informative, valuable lecture. My question is, if there is any side effects on plant tissues and the crops after UV treatment, and what happened to the survived insects which not affected not affected or not mortality.
الذي حدث للحشرة التي نجت هذه
Well,
Is it had any acquired resistance against UV?
هل كان هناك مناعة مكتسبة ضد الأشعة فوق البنفسجية؟
So that's a very good question. And one of our objectives from the beginning was to make sure, we wanted always to make sure that the UVC dosages that we apply are not going to damage our crop. And we tested different species to make sure that this is not going to have any effect on the cosmetic of it. And the result was okay, except for 2 species that showed a little bit of damage. And although those 2 species also could recover after 3 weeks, we didn't have any problem. In terms of making resistance, developing resistance in insects against UV, that's something that we need to consider. And it really depends on the dosages that we are using. So we are kind of like careful about like the amount of dosages that we are going to apply and to avoid any possible resistance. So yeah, I would be happy to answer any question. I'm leaving my email address here, so you're so welcome to shoot me an email if you have any questions.
Thank you so much.
You're welcome. Thank you.
Thank you دكتور مريم.
You're welcome. Thank you.
في أسئلة تانية يا جماعة؟
أرجو دكتور مريم المشتركين مع حضرتك في البحث هيقدموا البحث ولا حضرتك قدمتيه ليهم 3؟ Please ask here.
دكتور مارجريت are you here؟
Yes, دكتورة نهى.
لا دكتور مارجريت اللي جاية مش دكتور مارجريت حضرتك.
سوري
حصل دكتور مارجريت سكانر.
اوكي
دكتور مارجريت are you here?
So if you mean Dr. Skinner, she's not here. I don't think
Okay. Dr. Bruce?
And no, they actually asked me to make this presentation. We all are part of this project, but today I'm here to present the results.
Okay, you're welcome. طب في أي أسئلة تانية يا جماعة؟ Any question? Okay. Thank you so much, Dr.
You're welcome. You're welcome.
Doctor,
okay, thank you.
Dr. Ayman Yusuf have a question.
شكرا يا باشمهندس سعيد وشكرا دكتورة نهى I need to ask about the side effect of there are any side effect on
أعتقد دكتور أجرين خرجت.
أنا قفلت عليه؟
خرجت خلاص.
لا أجرين موجودة.
لا أنا بتكلم على
الميكروفون بتاعها اتقفل.
لا دكتور أيمن موجود. هو اللي بيسأل.
موجود يا باشمهندس سعيد ازي حضرتك؟
أهلا وسهلا اتفضل. هي الدكتورة
لو خرجت خلاص مش مشكلة.
اوكي
شكرا.
الحمد لله كده النهارده تم الانتهاء من تاني يوم من ورشة العمل الدولية، وبإذن الله نشوفكم بكرة الساعة 1 إن شاء الله لثالث يوم وآخر يوم في الورشة بإذن الله. السلام عليكم.
Thank you so much.